All right. Good morning. So I'm going to talk about, obviously, lionfish. We're here to talk about today. So I couldn't be there in person. Title of this is Invasive Lionfish in the Northern Gulf of Mexico, Population Trends, Ecological Impacts, and Potential Mitigation. And just want to acknowledge co-authors Dave Shigeris, Kristen Dahl, Holden Harris, Joe Tarnecki, Maya Taylor, Rob Aarons, and Mike Allen, all of whom are or were here at University of Florida. So invasive species are unfortunately very prevalent in the U.S., especially the southeastern U.S. Two main factors play into this. One is the amount of global trade that the U.S. does. Secondly is our temperate to subtropical in Florida environment. And so lots of species get introduced, but what truly makes a species invasive is it has to be introduced, established, and then spread in its non-native range. And then lastly, to cause ecological harm. So that last component, ecological harm, is really critical when trying to decipher whether a given introduced species is actually invasive. So in Florida, in the marine system, especially in freshwater as well, we have lots of non-native fishes that have been introduced, sometimes persist in the system, and occasionally, like the regal damsel on the bottom right, spread. But whether they actually cause ecological harm, in most cases, the answer is no, or at least that harm is not very substantial. What are factors that promote the spread of invasive species? So these include things like release from parasites or pathogens, which they have in their native range. Also, predator control is diminished. Oftentimes, predators don't recognize them as prey. This is especially true for a fish like lionfish that looks so different than native prey items. So they're not recognized as food. Another factor that enhances their ability to spread is that many of these species are opportunistic species. The analogy among mammals would be mice, and that they grow fast, they reproduce at young ages, and they reproduce a lot. So this removes some of the constraints that they may have had in their native range, and they can respond to favorable conditions quite rapidly. And lastly, they often have some type of predator deterrence or prey capture mechanisms, which may be novel in their introduced range. And so among the species at the bottom here, a couple of reptiles and a clown knifefish, freshwater invasive in Florida, right? They all have these deterrence or capture mechanisms that are distinctive. And obviously in marine systems, red lionfish, the most successful marine fish invader, they also have many of these factors. Excuse me. They were first spotted off of Southeast Florida in the 80s. It's actually a two-species complex, although most of the DNA suggests volitans is by far the more prevalent. They became established into the Southeast U.S. in the 2000s, Caribbeans by the mid-2000s, and the Northern Gulf of Mexico by the late 2000s, right? First fish in the Panhandle were observed in 2010, although otolith aging data from my lab shows they were here at least in 2008. And in the wake of their population increases across this invasive range, we've seen declines in prey species, declines in biodiversity, and effects on native predator-prey dynamics. This curve is produced by the U.S. Department of Agriculture, more for plant invasive species, but it presents what's called the invasive curve. And so early, when a species is absent, it's the best time to put effort into avoiding infestation and control costs by just preventing them, right? An ounce of prevention's worth probably $1 billion of cure in the case of many invasive species like lionfish. Early in the invasive time period, so the population growth is exponential, then it becomes linear, and then it becomes asymptotic as it reaches carrying capacity. Early on, there may be hope for eradication. Then we switch to a containment strategy. And then long-term is just protecting assets as best as possible. So we don't want to be in the red zone. And unfortunately, we're in the red zone with red lionfish. And so what drives this curve? What are the ecological underpinnings of it? Well, on the left, we have a function that's this S or logistic shape. And this is the logistic population growth curve. And so on the y-axis, we have population size, and on the x-axis, we have time. And then this dotted line toward the top is carrying capacity. So this is the maximum biomass that the environment has, in this case, if we're talking about lionfish. Now, carrying capacity can fluctuate because environmental conditions fluctuate. But in this case, we're just talking about a static condition to explain some of the processes here. So again, early in invasion, we have this exponential growth. Then the population growth becomes more linear. And eventually the curve heals over at the top of the S and becomes asymptotic as it gets closer and closer to carrying capacity. Well, why does this happen? Why do we have fast growth early on and then growth slows and then eventually the population is not growing at all once it reaches carrying capacity? And it's because of what's called density-dependent population growth. So factors that affect the size of the population or the growth rate of the population can either be density dependent, which means they're a function of the population size, or density independent. So things like wildfire would be density independent, or large-scale climate events. An asteroid hits the planet, right? Those are density independent. They have impacts that are irrespective of the density of the organisms. But then we also have density dependent, and that's what's affecting this growth curve, population growth curve on the left. And so the rate of population increase, also called R, is maximum when the population size is small, and then it declines as population size increases. So if this wasn't the case, then populations would expand infinitely and then crash. So we have great evidence from many classic studies, more modern studies, that demonstrate different types of density dependence within populations. And that's what causes this reach toward the carrying capacity and the shape of this logistic curve. Well, invasive species, they often have a boom period, which is early on in that invasive time period, and then the population growth becomes more linear, and then it reaches an asymptote as they reach the carrying capacity, peak in, or population size. But then a crash often follows, right? And then perhaps a new post-bust equilibrium is reached. And in some cases, we have these boom-bust cycles that are established, and there's some well-known examples of these. Introduced European hares in Australia, for example, where we have these boom-bust periods where there's a long period of low population size, conditions become optimal, population size ramps up very rapidly, and then it crashes again and sort of oscillates between these two. So there's been sort of a standing question about red lionfish and which of these two, if either, right, we may just get to a boom and it stays there, would they actually display? So we had a long-term data set that started before Deepwater Horizon in 2008 and 2009 at a series of natural and artificial reef sites in the north central Gulf of Mexico. So these sites then were visited with GOMRI, Gulf of Mexico Research Initiative funding, for 6 years quarterly, and then we've sampled them periodically since then. Sampled with remotely operated vehicles, you can see in the bottom right. So we can fly these down to the reef, we can do a quick survey, and then we're off to our next site. We work with charter captains. And so charter boat captains, obviously have a great deal of experience holding the vessel on site. And so we utilize them to go to our reefs, do the sampling, and then move on to our next position. In the top left, you can see a video from one of the Okaloosa Sea artificial reefs in 2016, and you can see just the massive number of lionfish. Many of you in the room have been in the water on these reefs and seen this firsthand, but just a tremendous number of lionfish. There were over 100 lionfish on this one artificial reef module. And so over time, because of this funding that we had to examine the effects of Deepwater Horizon, we were also able to map out and track the lionfish invasion in this region, maybe as comprehensively as any region studied, because we had data from before, and then we were able to watch lionfish move through because of this intensive oil spill related funding. And we see, rapid increase from 2010 through about 2014 or 15 in this region. And then we have an asymptote that's reached. There's some variance here, but these are the mean densities. And you can see at the top we have natural reefs and the bottom artificial. And something important to note is that the density of fish on natural reefs has been and remains about 2 orders of magnitude lower than artificial. But also on natural reefs, detectability is lower. So this is probably an underestimate of true density, but not by 100%, maybe by 10% on the natural reefs. But on the artificial reefs, you can see an asymptote was reached, very rapidly. If these fish were first here in 2008, by 2014 we've reached peak density on artificial reef sites. But these values of around 35 fish per 100 meters squared are the highest densities in the world for invasive, or not just invasive, but even natural populations of red lionfish. So what have been the impacts? Again, many of you dive and you've seen these firsthand. But lionfish, they prefer small demersal reef fishes like this juvenile damselfish or the 2-spotted cardinal fish, newly settled vermilion snapper. Down here we have bank sea bass loaded up in the gut of a lionfish. But diet research done by Kristen Dahl shows that they have the pretty generalist diet. They can switch to benthic invertebrates when demersal reef fishes aren't present. They can even switch to feeding on pelagic scads and anchovies when prey aren't available. So they can actually shift their diet and impact many different trophic levels in the food web. We've also seen this negative correlation between red snapper and lionfish. Now, correlation doesn't mean causation, but we've done studies with telemetry where we showed that lionfish, when they're present, red snappers stay farther away from artificial reefs where lionfish are present versus ones where they've been cleared, and they stay higher in the water column. So there is this negative interaction, and it likely has to do with the foraging halo of lionfish around the reefs and the fact that red snapper also are feeding on non-reef prey. So if lionfish are foraging down the reef prey near the reef and then a halo around the reef, it's causing red snapper to have to go farther to find a meal. And after a while, they just leave, or they leave at a higher rate or more quickly at sites when lionfish are present versus ones when they're not. We've done quite a bit of work on ecosystem modeling to examine these effects. I don't really have time to get into details here, but there are a couple of great papers by Dave Shigaris. The animation that you see on the right was produced by Rob Aarons and shows an an ecopath model of the northern Gulf of Mexico food web, and then utilized to examine the impacts of lionfish in the system, and particularly for reef fishes that are exploited. And the presence of lionfish means that some component of the prey base that would be going to things like red grouper and gag and red snapper ends up in lionfish biomass and production. And so there's this indirect effect, this between species competition, which even though lionfish aren't eating red grouper or gag or red snapper, at least at any high percentage of their diet, they do have indirect effects on those species as well. Well, what about density-dependent mechanisms in lionfish, right? We've seen the population reach an asymptote and then actually drop a little. So what are the mechanisms? Well, first we have density-dependent lionfish growth. This was a paper done by Kristen Dahl a couple of years ago, and it shows the otolith sections down below show opaque zones that we use to count the age of or estimate the age of the fish, just like you would age a tree by counting tree rings, but now we're counting opaque zones and otoliths instead of rings and trees. And so we can estimate the mean size at age across a range of different densities. So we went down and cleared lionfish off of reefs, and then we aged them, and then we could, because we had the reef identified, we knew how many fish were present. And so you can see each of these lines go from higher size at age, and this is for ages 2 through 4, females and males. And you can see that there's a substantial decline in size at age. Males are bigger than females, but in all cases, the slope of these lines is very similar. And so when we get higher densities, we're getting this negative feedback. It's density-dependent growth feedback in individual fish. So clear evidence of density dependence, at least in growth. We've also documented extensive cannibalism in lionfish. Now, diet studies often fail to detect the presence of lionfish prey in the stomachs because most often we have these fairly small prey items. So we have this fish at the top. It's about 3 millimeters long, or just a little over an inch. So it's a newly settled juvenile. Same with the one at the bottom here, that were taken from lionfish stomachs. So we rarely see them in stomachs, but we do get unidentified prey, which we use a technique called DNA barcoding to estimate the species of the prey item. And then if it comes back as lionfish, then we can do genotyping to test whether it was a different tissue or DNA, if the DNA was different between the prey item and the consuming lionfish, because there's a lot of discussion about the potential for contamination. If the prey item in the stomach is too degraded, then perhaps you're only amplifying the host, or excuse me, the consuming lionfish's DNA. But by doing the genotyping, we can actually determine that no, the prey item in the stomach is lionfish, and it's different than the DNA of the consuming lionfish. So it's cannibalism. And so this extensive cannibalism would have a density-dependent feedback on recruitment. If these animals are feeding more prevalently on their own recruits at higher densities, then that's another type of feedback mechanism that exists. Well, another type of density-dependent population regulation is often disease, especially if disease is affected by transmission rates that are determined or affected by the density of animals. So when you have higher densities of animals, you get higher incidence of disease transmission. I mean, we saw this recently with COVID-19 in big cities and how quickly it could move throughout the population there because of higher contact rates between individuals. So we have this ulcerative skin disease. I know Teresa is going to talk about this in more detail next, but I'll just briefly say, you know, this first appeared in 2017. To date, we have an unknown etiology or the causative agent. Again, Teresa may add some new information with respect to that. And then there have been significant declines in lionfish condition, as well as population density after the disease was present. The map in the bottom left shows everywhere that the disease had been reported, at least in 2018. And this is a paper by Holden Harris. And the next slide shows the density of animals in our region at artificial reefs off of Okaloosa, Escambia, and then natural reefs in the system. And so what we can see here is that we have substantial declines at high density and low density artificial reef sites. So the low density sites were sites where we had earlier removed lionfish and we were looking at how quickly they can recover their populations. But we see these substantial declines in both systems at about 80%, 75 to 80% on Escambia artificial reefs, Okaloosa artificial reefs, and then natural reefs in the north central Gulf of Mexico. So this did have a substantial population level effect. And if we look at our estimates of density at artificial reefs, you can see that drop in 2018. And then as we move forward, we haven't been able to sample quite as extensively, but we did do some summer sampling on these artificial reef sites in 2019 and 2020. And the red lionfish densities remain quite low. And then data for 2023 shows a slight increase, but still quite low relative to the earlier maximum levels of population biomass. So is this the new norm? Is this going to be what the population level is? Now, I know that the commercial fishery has had issues after this decline, and some folks have stopped fishing, spearfishing for invasive lionfish. Some of the spearfishermen we work with indicated in the past couple of years they saw much higher densities in certain regions. We don't see that in our data, But then in 2024, or excuse me, in summer of 2023, another outbreak of the disease knocked back those densities again. So maybe the level of sampling that we've been doing in recent years, we're missing these pulses in population increase and then drops due to disease prevalence. But again, maybe Teresa will enlighten us a bit about the disease dynamics. So again, where are we? Are we at a new post-bust? This is the new normal? Or are we in a cycle of recurring population boom and bust? Again, the data that my lab has, we're not able to resolve this. It would be great to have the level of funding that we had previously to examine population trends over time, community dynamics. But that's, we don't have that. So we can't say with any certainty, but it appears we might be at the top curve and not the bottom curve. But, it still begs the question, how do we get all the lionfish out of the system? And the figure on the left is an experiment done by Kristen Dahl when she was a PhD student here at UF. And it shows the recovery of lionfish populations on Escambia East artificial reef sites after all the lionfish had been removed. And what it shows is that they're increasing fairly rapidly. And by 100 days, their densities are above 5 fish per 100 square meters, which is sort of a threshold in the literature about ecological impacts of lionfish. And so at least during that time period in 2015, 2016, when we had fairly high lionfish densities, they could come in from surrounding areas perhaps and recolonize rapidly. We've also done quite a bit of work on testing different trap designs in hopes of developing a commercial trap fishery. Unfortunately, even our highest CPU catch per unit of effort for the Giddings type of purse trap, not very effective overall in removing lionfish biomass from the system. It's more effective than either sea bass pots or lobster traps, but lobster and sea bass tend to be pretty effective in catching commercial reef fishes and other native reef fishes in the system, which we don't want to catch, right? The idea is to remove lionfish biomass to benefit the native reef fishes. We don't want to kill them as bycatch. So unfortunately, the trapping we've done to date doesn't suggest it's going to be, sort of a panacea, although trap modifications may help these become more efficient. Well, there's another potential approach that could be utilized, and it's referred to as gene editing. And there's an example in South Florida. It's been done in South America as well, where mosquitoes that are disease carriers like dengue fever have been edited. Their genome has been edited in the males by insertion of what's called a gene drive. It's just an edited segment of DNA. And that gene drive then is transmitted to the offspring. And by a few generations, all of the individuals in that region will have this genetic, it's basically a human-caused mutation, which then makes them sterile. They can't reproduce. And so this has occurred in South Florida. There have been recent results indicating that localized decreases in mosquitoes. This is very controversial and went through a large period of review before they were allowed to be released. But success has been shown in South America as well as now in the southern tip of Florida. We actually started to develop a gene drive for lionfish prior to the lionfish program getting substantial funding cuts at the state level. We've sequenced the lionfish genome, and now we're doing chromosome assembly kind of on our own dime. This is work with Dave Portnoy at Texas A&M Corpus Christi, work being done in his laboratory, with the idea of producing a gene drive for lionfish. Now, we're many steps away from the possibility of this, but it's something that we continue to work toward. Obviously, it would be very controversial to release a vertebrate with an edited genome to try to wipe out lionfish in the system. But right now, we're at the stage we just want to see if we can do it. If we can do it in the laboratory and make it work, then this is a potential solution to the problem. Again, really, really early stages of this development. So conclusions, right? Evidence exists that lionfish had reached carrying capacity in the North Central Gulf, at least on artificial reefs, by 2014, 2015. Density-dependent growth was apparent, along with prey switching behaviors, including cannibalism. Ulcerative skin disease transmission is likely also density-dependent. There's mixed evidence of lionfish population recovery following disease outbreak. Again, I can't speak to the anecdotal reports. I just can show you the data that we have. And so there's some evidence being discussed about increases, but our data from at least the sites that we've been studying doesn't show that, at least to a substantial degree. So there's a question that remains about what are the true boom-bust dynamics for lionfish, whether we're getting into an oscillating pattern or we're going to have a new lower density level. And then lastly, future control measures, right? Is there another creative solution? I mentioned gene drive. Maybe there are other solutions out there that haven't been proposed yet. Lots of folks to acknowledge and thank for help in the work, as well as funding agencies that provided resources to allow us to do it.
Thank you, Will. That was really good. We do have time for some questions. If anyone in the audience has some questions. In back.
Hi, have you guys seen any evidence of density-dependent control with behavior at all, such as like hunting success at different densities?
Did you get that, Will?
No, I didn't. Could you repeat it, Robert?
Oh, thank you. We'll give him a mic.
Hello?
Yes.
Have you seen any evidence of density-dependent controls with behavioral ecology, such as like hunting success at different densities?
You said hunting success? Is that?
Yeah, hunting success at like different densities, so behavioral ecology.
Right. So we haven't measured that directly. Spent time underwater doing it. But we do have some evidence from acoustic telemetry. We've done some three-dimensional acoustic telemetry for lionfish. And lionfish that are on sites that have higher densities, they spend time a little bit in the water, more than they do on sites where they're lower densities. And again, that matches the feeding ecology work that Kristen Dahl had done, that showed them switching to things like scads or anchovies in the water column, and also spending time farther from the reef structure on sites where densities are higher. And they have this really interesting behavior that they will occur, even 50 or 100 meters away from a reef, but they kind of occur in these long lines when densities are really high, at least densities that we saw back in 2015, 2016. So as long as they're sort of connected to their buddy, they'll forage away from the reef like that, but otherwise more clustered around the sites. So at higher density sites, they're farther from the reef. And when a fish is, even 25 meters away from one of these reef sites, the likelihood of catching a damselfish or a cardinal fish is really remote, right? So they're feeding on other things like benthic fishes or even pelagic fishes.
Thank you.
Any other questions? Well, that was a great talk, Will. And yes.
So you mentioned that you have had some funding cuts that have limited your ability to monitor lionfish recently. Have there been any efforts to use citizen scientist approach to kind of address that? I mean, we have a lot of divers in our area that I'm sure would be more than happy to assist with those efforts.
Yeah, we haven't done that specifically in my laboratory. Stephen Cyphers published a paper a few years ago about citizen science and lionfish. And I know that there are some dive clubs in the region, some of that are very active, that have long time series of lionfish count data. And so, yeah, the potential there definitely exists. We haven't explored that to date.
And we will have some talks later on in the agenda that may touch on exactly that. I know Rick O'Connor, were y'all able to do your dive last Sunday?
No.
At Fort Pickens? I wasn't going to go, but I see Brady here, you know, he's a lot more gung ho, but, any other questions? Okay, this time I definitely don't see any hands, so thank you again, Will, and we really appreciate your continued work on it. Sounds like, there's some interesting things on the horizon. The gene editing and the population control potential of release of those organisms does sound interesting and scary at the same time. So thanks. I appreciate your update.
Thanks, Robert.
Okay, we are a few minutes ahead of schedule, and Scott's getting Teresa Cody up and running. She's going to give us a talk on those cutaneous ulcers that Will referred to in his talk. I see her on the screen. Good morning, Teresa.
Hi, I'm just trying to get my talk up. All right,
We can see
need to
you.
share my screen. Hold on. That's it. How does it look?
Looks great.
It looks like, I mean, I can see
We see,
that weird, that part up at the top, which we don't want. Can you see that?
Yes, we
Can you
can.
just see the whole?
No, that's nothing you'd have to do, Robert.
Okay.
That is on her screen, I believe.
All right, let me figure this out.
If you change the display settings at the very top and just change that to duplicate, where it's the middle section says display settings
Display settings. Oh, here.
in the black box, yes. And duplicate slideshow. Should work.
There you
Okay.
go. All right, take it away, Teresa.
Okay. Well, also, I'm trying to
See your
get
notes.
rid of the participants.
Okay, we don't see that on our end.
Okay. All right. So this talk is based on our investigation of cutaneous ulcers that affected lionfish in the Gulf of Mexico and showed up in August of 2017. And these are the results of our study. They were published in the Journal of Aquatic Animal Health. So you can see here, this is a long list of authors. The numbers go up to 9, and basically it's a diverse group with different interests, resources, and areas of expertise. And you'll see that Dr. Will Patterson and Alex Fogg are also on the list, both of whom are speakers at this workshop. So you all probably know a little more about lionfish than I do, but they are native to the Indian and South Pacific Ocean. They are non-native invasive species in the Atlantic Ocean, the Gulf of Mexico, and the Caribbean Sea. And you can see by this map just how successful they are. They are able to reproduce every 55 days, and they are voracious predators, and they can cause a lot of havoc on the ecosystems in which they are found, as you know. So anyway, relevant to this research, there were a couple of different groups out and conducting population surveys and conducting their research when they came across an epizootic of lionfish with cutaneous ulcers. So they contacted the health people they knew, and the health people contacted each other. And what follows is really just an ad hoc investigation of discovery into what might have caused the ulcers. So initially, we really didn't know about the extent of the epizootic, but we eventually got reports from the diving community of ulcerative fish on the Atlantic side of Florida and in the Caribbean Sea south. And so for our study, fish were netted and speared and brought onto the boat and examined for ulcers. And one of my collaborators gave me this picture of this guy collecting lionfish. I don't know who he is, but if you're in the audience, just want to say hello. This is a map of the monitored reefs. And so you can see white circles, black circles, and red squares. Well, the white circles are reef sites where no lesion fish were found. The black circles represent reefs where lesion fish were seen, and the red squares are areas or reefs where fish were collected, both normal and lesion fish, for necropsy by our groups. So at the end of the day, we looked at about 200 fish, both normal and healthy, and what we did with them really depended on when we got the fish post-mortem. A lot of the fish we got on the same day, but some of them were as old as 72 hours old by the time we got. So really, you can't do a lot with those fish that are 72 hours old, but we were still able to get some information from them. So these are just some of the things that we did, internal and external evaluations. We took measurements and we weighed organs to get a look at some of these health, non-specific health indices like condition factor, hepatosomatic index, and splenosomatic index. These are important because they are a reflection of the nutritional and energy status of a fish, and they can change depending on what's going on with the fish. But we also accounted for other variables that could affect these numbers, including the year, the location, the age of the fish, and sex of the fish. We fit a two-way ANOVA, analysis of variance, with to determine if there were differences between normal and ulcerated fish based on their age, or if they were male or female. And we looked at a variety of tissues to look for pathological changes and for the disease, like perhaps parasites or bacteria. We examined the microbiome. You've all heard a lot about that in the news lately. It's basically just the community of bacteria that you find on your skin or in your gut or wherever in the dirt. And I know that we looked at 10 ulcerated fish. Unfortunately, we did not do this for normal fish, but that's something that maybe we could look at in the future for future research if this thing rears its head again. We did do lionfish prevalence surveys, and the data that was included in our paper started in August of 2017 and concluded July 2021, though I know that these surveys continue to this day. So in summary, we looked at some variation of using diagnostics for 70 normal fish and 66 ulcerated fish. We did bacteriology on several of the organs. We did a microbiome analysis to look at what was present on the skin. We looked at these various morphometric health indices, and we did gill and skin squash preps to look for parasites, and then we looked at the histology to look for any kind of pathological changes. So the results. All right, so condition factor was on average 7% lower for ulcerated fish. And that can just show that something is going on with the fish. And we also looked at this hepatosomatic index, although it was not a statistically significant result, it was 8% lower. Splenosomatic index was on average 80% higher for ulcerated fish, and that can be a sign of splenic enlargement, which is kind of a sign of immunological response to a disease. So we think that's pretty relevant. If it was male or female, it didn't really matter, but we found that fish were on average a little over a year older, the ones with ulcers. What this means, we're not really sure. So this is generally what the fish look like. I'm sure most of you know that, but just in summary, there was no real specific location that the ulcers were found. They could be found anywhere on the body. You could see like 1 to 3 ulcers on the body, and they all had the same pink, shiny red appearance. Otherwise, the fish really didn't appear to be sick otherwise. You can see that they had really good body condition. Even though the condition factor was lower, they still looked pretty healthy. They didn't really appear to be emaciated. We did do those squash preps, and there was nothing really informative from that, except that we did find that the clinically healthy fish did have a higher level of parasites, normally trichodinid parasites, but parasites in fish are pretty common. So about 6 months after the start of the epizootic, the field researchers began to see fish that looked like these in the field, and they had pale patches of skin, is shown on the right, on the left, and these distorted pigment patterns, which they categorized as scarred. We were able to look at a couple of these in the lab, and the histology was consistent with healing, wound healing. This is what some of the histopathology looks like. I know it's kind of, you may not know what it means necessarily, but this first photograph, these are representative sections, first of all, of what the histology looked like from the ulcers. So the first A shows skeletal muscle with underlying the skin. And then B is a little bit higher magnification. So you can see these granulation tissues along with the skeletal muscle. Well, in a normal healthy fish, you would expect to see just this skeletal muscle, right? But these granulation tissues are a sign of healing. It's like fibrotic tissue, which is basically connective tissue. And if you go over to photographs C and D, you can see newly formed blood vessels, fibrosis, and leukocytic infiltrates, which leukocytes are typically a response to some kind of an infection or disease. It's part of the immune system. These arrows show some necrosis in the muscle tissue, which you would expect to see, but overall, this picture shows that fish are healing. We did look at other organs. This is the kidney, and this yellow circle shows some necrosis. And we looked at spleen, another important organ, and that's, remember, it was enlarged, the spleens were enlarged, and that was a significant finding. But here in the circles, you can see necrosis, and then you see vacuolation. And this vacuolation is pretty consistent with histiocytic macrophages clearing debris. So definitely, signs were healing. These are melanomacrophage centers. Again, those are, it's a host response to something. The bacterial results were pretty inconsistent, but what we did find most commonly was this Vibrio harvii. This is a known opportunistic pathogen. And so even though we had inconsistent results, it still showed up the most. Each number is an individual fish. So there were sometimes fish where only Vibrio harvii was isolated from one of the organs, or sometimes there were multiple species. Vibrio harvii are known to be isolated in mixed microbial communities. So, that's not anything that's really surprising. But you can see how we isolated it from numerous organs. These in the kidney and the liver are more meaningful than something that you might find in the ulcer, just because, when you're conducting the sampling for isolation, sometimes you might get contamination. We don't know that if this was after the fact or not. One of the things, that's notable is your skin is one of your innate immune properties. It protects you from stuff getting in there. But obviously, if you have an ulcer, there's a portal of entry for bacteria. So it's possible that some of these isolates were after the fact, but we don't know. All right, so this was the microbiome, and it's pretty busy, but again, and you can see that a lot of different taxa were identified. However, this, remember we had 10 fish. Oh, how do I go back? Okay. But what you'll see, what stands out the most is that Vibrio harvii was the most dominant species, followed by another Vibrio species here in pink. And then the other notable thing is that these bars are green, but these simply represent others. And it really means that those numbers were so low that they weren't identified. I think the biggest thing to look at, though, is the fact that Vibrio harvii was most commonly dominating. So regarding the prevalence surveys, you can see here that prevalence was highest at the inception of the epizootic, and then there was a precipitous decline followed by these small spikes. And it looks like they normally occurred in December and November. You can see that here again. And there was a big drop off here in 2021. So why didn't we identify a communicable pathogen? It's possible that the methods we used may have not been effective in identifying the pathogen. Maybe the bacterial culture conditions weren't right, or the media we used wasn't specific enough. Only like 1% of bacteria are culturable, so we could have missed it. However, you think that you would find that really in some of the histopathology slides. There were no acute lesions in the fish that we sampled. Remember that healing was occurring, it was evident, and so maybe the acute disease had no clinical signs. The acute phase in the disease process may have been really a very short duration. It's very possible that the disease didn't have a communicable etiology. Ulcerated fish showed up simultaneously across the Gulf of Mexico and in the Caribbean Sea and on the Atlantic side. So it wasn't like a coral disease where you have where it starts and then expanding out and spreading from that point. There really was no ground zero. And research does exist demonstrating the occurrence of ulcers appearing spontaneously with stress. So it's possible that there really wasn't a communicable pathogen. But in the future, for this sort of surveillance, we thought about what we could do a little bit better. And I caught the tail end of Will's talk, and it sounded like there was a decrease in funding for monitoring, but ongoing year-round population monitoring is helpful, of course. Monitoring the entire fish community, not just one species, is helpful. However, I really believe that if they had seen ulcerated fish of other species, we would have gotten them and heard about it. It's important to collect baseline data on the key species, such as the morphometric health indices, looking at just basic pathology or histology, understanding what's normally present, examining the microbiome so you can see changes in that might result, and biomarker assays. As I mentioned, this was sort of an ad hoc response to an epizootic, so there wasn't really a standardized protocol for this particular research study, but maybe that would have helped make things a little bit smoother. I don't know. I feel like at the end of the day, we did enough to find this potential pathogen or the cause of the ulcers, but we just weren't able to find it. Finally, I think that the data should include some environmental parameters. We didn't have that consistently for all the sites, but YSIs are expensive, and I just think that if we were better mobilized, maybe we could have gotten some of that. But at any rate, in addition to all the people that worked on it and were authors on the publication, I think we all know that a lot happens behind the scenes. And so I'd like to thank our histology people that gave us such excellent histology slides, other people in our group who helped do the necropsies. We presented some of the pathology results to other pathologists, and they weighed in a little bit, aged the fish. And of course, we do thank our citizen scientists, Bob and Carol Cox. We got quite a few specimens from them. And it's people that are really out in the water that can notify us that something is wrong. So I heard that I had a few extra minutes to talk, and you guys are ahead of time, but this is just a kind of a quick overview of my program. So hopefully if you see stuff in the future, you'll give us a call. We have a few components in addition to responding to and documenting fish kills and epizootics. We also monitor health in fish across the state. We conduct disease research, such as the study that you just saw, and we work on developing baseline health profiles for different marine sport fish species. That's partly in support of stock enhancement. You want to be sure that whatever you release out into the wild is not going to negatively impact the wild populations. So we have field labs all over the state, and our fisheries groups, they go out and they, independent of anglers, they go out and they collect population data, our fisheries independent and dependent monitoring data. And if they see any fish that are abnormal or diseased, they send those to us and we conduct necropsies. Sometimes we can figure out what's going on, and like with the lionfish, it remains a mystery. The other way that we get our specimens is through our marine fish kill hotline. My group oversees that. So there are a couple of ways to report. You can either do it on the marine toll-free number, or you can submit a report online. This was established in 1995, but we do have records that go back as far as the 70s. So we really have a database with decades of disease data and fish kills and that sort of thing. So it's a good repository for this kind of information. So we really do like to try to get as much as we can so we can see any changes in the environment. So, like I said, although we document and monitor aquatic animal disease, I mean, that fish kills are primary, but we get calls about all sorts of things, invertebrates and turtles, bird kills, that kind of thing. And it's really not just to give us information, but we provide information to the public. We're a state agency, so we provide information about parasites and pathogens and what might cause fish kills. So it's a resource for the public as well. We do have a pretty informal network for response. We work with the DOH, the public, local and city government, academia, water management districts. You know, we don't oversee everything, obviously. We work with the turtle stranding group, marine mammal lab, and that sort of thing. We have a variety of diagnostic capabilities, as you saw from the talk about the lionfish, necropsy, histology, microscopy. We can conduct electron microscopy, microbiology analysis, along with the molecular and parasitology. And this is just one of our cool cases. This is a lemon shark, and you see here all these trails of silver. But when you take a look at it a little bit more closely, this is taken on a dissecting scope, so you can look and see what that is exactly. This was seen under a compound microscope. If you look really closely, you can see that these are encapsulated nematodes. And when you look at the histological slide, you can see it in the skin of the shark. These are just some of the other cases that we've seen over the year. We get hundreds, sometimes thousands of calls in a year. Our group is small, so but we do respond to every single report that we get. We try to call everybody back. This is lymphosarcoma and redfin needlefish, which was primarily seen in the Indian River Lagoon. And that came about from just years of monitoring from our fisheries independent group. So they would go out and they would sample, and, you know, at some point, one of our staff said, you know, we're seeing these a lot. And so then went back to look at all the historical records and see that there was actually a lymphosarcoma in these redfin needlefish in the Indian River Lagoon. And really, that's the only area that we've seen it. We haven't seen it, you know, so much in the rest of the state. One of our colleagues got a grant to look at disease in amphibians and was the first to find ranavirus and co-occurring infection in frogs in Florida. These are just some other examples. These came in, these shrimp, they have a disease called cotton disease, and they're really just caused by these microsporidian parasites. Here, these fish were submitted to us by Escambia County. They were a little bit concerned, the people were concerned that fish were tumorous. And so we said, send them to us. We looked at them and found that they weren't actually tumors, but they were myxozoan parasites. In addition to those case studies, we also, like I said, develop baseline health for profiles for these different species. This is a Goliath grouper that was obtained after those cold snaps in 2010. So we were able to get, even though fish kills occur, it's actually an opportunity to get health information and disease information on some of these species that we normally aren't able to get, because we also are not allowed to collect Goliath grouper. We look at offshore reef fish. We've done some evaluations on blue crabs. Red drum is our hatchery fish. And so we've looked at a lot of different fish over time. We provide input for managers. And this is just a case where a lot of people were getting sick. Back in 2005, they were exhibiting neurological symptoms. and they had tingling in their fingers and their toes and vomiting. And so what we found was that they were harvesting, these people were harvesting pufferfish, and those pufferfish had sequestered saxitoxin. We looked at other areas in the state, like Tampa Bay, for example, and we didn't see that same level of saxitoxin in the muscle tissue. So a ban was put in place only for the Indian River Lagoon and nowhere else in Florida. But it was our group that helped work on that in conjunction with the harmful algal bloom group. So, I mean, we really don't work alone. We collaborate all the time. This was just I think it's one of my last cases, but this was back in the 90s. There were fish showing up with these really horrible lesions. But as you can see, it's not just, those were really in St. Lucie County over here, and over here, just in the Caloosahatchee River estuary. And so that's kind of how it all started. But then we found that this, it's a freshwater oomycete that causes it. It's kind of like they have hyphae, fungal hyphae, which you can see here. You can see that it's actually found all over the state. But one thing about this freshwater oomycete, I mean, they are in the estuaries here, but it's really what occurs when there's an influx of fresh water and the temperatures are a little bit cooler. So we tend to see it more often in Northwest Florida when the weather is cooler. And in fact, we just necropsied a few fish, I think up to 6 fish yesterday, fish with aphanomyces and Baden's infections. And one of them was a mullet, and it was so bad that all the muscle was gone and you could see the organs. So it was pretty bad. These are granuloma formations, and it's basically host response to wall off these fungal hyphae. Real interesting. But they were also seeing it up in the Northeast as well. All right, these are just a couple more. These are Amlodinium ocellatum on red drum gills. Those can increase to real dense levels and cause problems with respiration for the fish. This was kind of a cool bacterial pathogen. It was an Aromonas species that we isolated from a white catfish kill. And this is a blood auger. And you can see this clearing around each of the colonies, which tells you that it's hemolytic bacteria, which means it lyses blood cells to get the nutrients. And then we see like gross parasites, macroscopic parasites that we get from the public sometimes. So having said all that, I'll take any questions. Hopefully I didn't go over time and didn't really monitor.
Thank you, Teresa. That was really good. We do have plenty of time for questions. Chantil's going to provide a microphone.
I was just curious, spearing lionfish myself and watching other people spear lionfish, it's not unusual for them to get away because you don't use a barbed spear. So I'm just wondering if there could be any connection to ulcers with a fish that's speared but then gets away and sorts injured.
It was so widespread and we had never heard about it before that we considered that as well, but we didn't think that was the case, especially after we examined the fish in the lab. It's not like there were holes in there. And like I said, they were typically healing. So I don't know if Will has anything else to say about that, but we didn't suspect that it was due to being speared.
Okay, and Laura's got a question.
Did you ever make a determination whether those fish were still able to be consumed if they had the lesions on them?
Well, first of all, we're Florida Fish and Wildlife, not the Department of Health, so we never ever provide human health recommendations. However, I mean, as a general rule, I would think that you probably wouldn't want to eat anything that looks like that. As far as the fish that were scarred, I mean, it was just the histology showed that they were healed. You know, the sections were consistent with wound healing.
Mark has got a question. Oh,
This is less of a question and more a couple of comments. I had about 200 or 300 lionfish alive in a lab for about 2 years. And we noticed a lot of things. He was asking about spearfishing, and we have a lot of very healthy fish come in with no holes on them and everything. And within 2 or 3 months, they would develop these ulcers. So the spearfishing probably wasn't making it any better for these fish, but it wasn't like the ultimate cause. We would have completely healthy fish that would develop this disease in captivity. You had a comment on one of your slides that it's potentially that the acute phase of the disease was why you didn't really see any statistical significance on the condition changing. I think you had a slide that said it was about 7% different. And we found something really similar where it was from onset of ulcers to death was almost always within 3 days. So they didn't really have time to lose a whole lot of like muscle mass or body mass or anything like that. And the only thing that really delayed it at all was really low hyposalinity and low temperatures would keep them alive for another couple of days. But we threw like every sort of erythromycin, antibacterial, antifungal stuff at them, and none of it ever cured any of them.
Yeah, you're right. Now that you mention it, we did get reports of from a woman doing her thesis, and she said the same thing. Like, she was holding these fish, they developed ulcers, and then some people say that they died. I know that Dr. Hawk at LSU also had the same thing happen. So yeah, definitely evidence that the ulcer formation wasn't necessarily due to fishing.
So Dr. Hawk was
Activity.
actually my fish. We were giving them to Dr. Hawk as soon as they were dying. We had a lab just across campus that we were giving to him, and he was culturing them for like gram-positive, gram-negative, everything like that. Found the same thing that you did, just a bunch of Vibrio, which is, I mean, that's
What did they find? Did you say
just a
Vibrio?
bunch of Vibrio species, which was already in the water anyways. It wasn't really super helpful, but yeah, Dr. Hawk was the fish that we were using.
Okay. Yeah, we did talk to him briefly about that too.
okay, Mark Nicholas has a question.
Yeah, I was just wondering if you did a literature search to see if it occurs in the native range Indo-Pacific, any reports?
Repeat that, Robert. I didn't hear.
Sure. He was asking if you did a literature search, if there's any of these ulcers that occur in the native range.
We didn't find it at the time, but that doesn't mean something hasn't shown up since then.
Okay, we have a few more minutes. I actually had a question. It was more related to the baseline data that might help us elucidate a potential problem pathogen in future ulcers, and that is the way that we typically collect lionfish is they're speared and they're put in a zookeeper, and there's a lot of contact with their other fish that are in the sample. So if we were to want to conduct any research on the natural biome, microbiome on the skin in particular, that the protocols that you mentioned in one of your conclusion slide might be isolating, putting those fish in plastic bags and isolating individuals in a collection so that we could get a better handle on what was on a fish as it was swimming versus what was transferred from fish to fish in a zookeeper or cooler. Not sure if you have funding, if you would be interested in receiving fish that were isolated like this in the sampling protocols, but just wanted to mention that to you as well.
So regarding that, the fish that we did have microbiome data from were collected when we went out into the field, and the fish were brought up and sampled right away. I think we had one fish that came from the researcher that I was telling you about. But the fish was a couple of days old by the time they got it for analysis. And so we couldn't really use it because you don't really know how the microflora changes from the time that the fish is collected to, several days later. But that's something that has to take place immediately. Like, they're pretty stringent about collections for microbiome analysis.
Okay, any other questions, comments? All right, we are a few minutes ahead, but we're going to go to break. Thank you very much, Teresa. We appreciate that.
All right. Thanks for having me.
And we will be on break until 10 o'clock, and we'll continue our research updates, part 1. So stay tuned. Thank you, Teresa.
We're going to go ahead and get started on the next little session. I know on the agenda it says research updates part 1, but obviously this is part 2. My name is Thomas Dervis. Thank you all for coming again today. Today, the first little session for the research updates part 2 is with Dr. Alexis Janicic. It's the detection of lionfish in Northwest Florida using molecular tools. Dr. Janicic, it's your turn.
Good morning. Thank you, Thomas. It's so nice to see so many former students in the room. So happy to be here today to talk to you about lionfish work that we've done in my lab with many collaborators. And I'll start in Northwest Florida, but then I'll move a little bit around the Gulf and the Caribbean as well to tell you some work that we've done with lionfish. But the theme here is that we use molecular tools, and that's what we do in my lab at the University of West Florida. We're mainly a molecular lab. And so if it has a molecular component, we're excited about it. So it can be on salamanders, it can be on birds, it can be on fish. And in this case, we've done some work on lionfish. So I'm going to try to tell you 3 quick stories today. So I'm going to go kind of light on our molecular methods and more about the stories. So the first story will be about invasive lionfish detected in estuaries in the northern Gulf of Mexico using environmental DNA. Our second story will be about missing the mark or missing the marker, how we identified pseudogenes through mitochondrial genome sequencing in lionfish. And then also characterizing the diet of that fish, or that bird rather, the least tern. I mean, I'll tell you some work that resulted in lionfish results for that bird. Okay, so our first story is about invasive lionfish in the estuaries in the northern Gulf of Mexico. So this work was published in Environmental Biology of Fishes a year or 2 ago. This work was done with former graduate student in my lab, Amy Brower, and former postdoc, Justine Whitaker. And so we wanted to look into the estuaries in Northwest Florida from an environmental DNA perspective, because we had heard that lionfish were in estuaries. They were observed in 2010 near the Jupiter Inlet. And then also in 2011, they were documented in estuarine environments in the Velar Estuary. So way on the other side of the world, but they were still in estuarine habitats. And given our prevalence of lionfish invasion here on the Gulf Coast, we wanted to see, were they invading our estuaries and river systems. So that was kind of the start of this project for this work. So as you know, estuaries are important nursery habitats for many fishes, including juvenile and economically important species. And lionfish, as you know, are not caught on hook and line. And they're also hard to do a visual survey for in an estuarine environment, because typically estuaries are murkier and the water is not clear, it's more turbid. And so just seeing them from a visual perspective is hard to do. So given all of those sort of facts, we decided to come up with a solution to sort of investigate those lionfish in that area. And so we used environmental DNA. So we've heard already about lionfish and why they're such a problem. But in our area here, particularly in those estuaries, that provides the ideal breeding temperature for them most of the year. So they can spread relatively quickly in this range. And then, of course, they compete with native species, right? So they prey upon the same organisms as native species. They prey upon juvenile species, which estuarine habitats are known for hosting, and they reach reproductive age sooner than native species. So, and they also produce way more eggs than native species. So all these things together make them, you know, a bad thing for our estuarine habitats, in addition to the Gulf of Mexico. And there's just a photo showing some gut contents of one lionfish, right? So tons and tons of little fishes are inside those stomachs. So given that background, our objective for this first research project was to see if lionfish had established residency in estuaries and river systems in the northern Gulf of Mexico with the use of environmental DNA. So let me tell you a little bit about environmental DNA first. You may have heard about this, kind of a buzz topic for the last 5 to 10 years. Oh, that's bad narration, hang on. So environmental DNA is basically DNA that's left behind. So in the case of fish, we're looking for scales or slime, feces, urine, gametes, or remnants that are in that habitat, that, you know, the fish swam through that habitat and it leaves this trace of DNA behind, just like if we think about forensics in a crime. All these fish are not committing crimes. And when we do this, the mitochondrial DNA is the most frequently used marker that we look for, because in a cell, there's lots of mitochondria, these little green things. I guess my laser isn't working. It doesn't work on a TV. Who knew that? Okay. So all those little green things are mitochondria, right? So there's many copies of DNA in a cell for that. Whereas in a nucleus, there's just one copy of DNA. So we use mitochondrial DNA because we just get a greater chance of finding that needle in a haystack, if you will. And then it's more stable in the environment and less susceptible to degradation. So that's another reason why we use mitochondrial DNA. And there's many copies in the cell. And this allows us to target a small fragment of DNA. We don't look for big fragments, although that'd be great, but DNA degrades relatively quickly, particularly in a saline environment. So we look for small fragments for that. So why did we choose eDNA instead of getting out there, you know, and trying to spear them or trying to snorkel for them? Where number one, it's hard to see in those habitats. But number two, we just dip in, take a little water. So it's efficient and it's non-invasive. It's been used for endangered, cryptic, and elusive species. My lab does a lot of work with environmental DNA. We've worked on highly endangered things like the Alabama sturgeon and the reticulated flatwood salamander and so on. So it's used to look for these sort of hard to find things. And sometimes conventional methods can be damaging, right? So sometimes you can get in with a seine or a trawl or things like that, and that can damage that particular habitat. And so this is particularly useful when small-bodied or cryptic organisms are difficult to sample, like in the case of our lionfish. And it becomes really powerful when we combine eDNA and conventional sampling. So when we put sort of these two methods together, becomes quite powerful. And of course, you don't have to have any direct contact with the organism. There's less gear bias and so on. And people have said that this is starting to reform the study of marine biodiversity in combination with other methods. So let me tell you what we did for this lionfish eDNA study. Okay, so we went out, we collected water, and then we extracted DNA from the water in all of these places that you see here on the black dots in Alabama and Florida. And then what we did was target part of the CO1 gene, which is like a barcode gene. It's a really common gene that's used for barcode studies. And we targeted 153 base pair fragments. So just 153 nucleotides, then we could say that was lionfish or not. What was cool and important to us about making these primers is that we wanted to only amplify lionfish, right? So we don't want to know if it's a redfish or a speckled trout. We want to know only if it's lionfish. So we made it really specific to that particular species. And then, of course, after we did those results, we sequenced it to make sure it was actually lionfish. Uh-oh. Am I frozen?
If you get the mouse and press the center of the screen, the right arrow should work.
Oh, okay, there we go. Sorry. Okay, so here's what we found. So basically, we found lionfish DNA in the estuaries and up into the rivers. So you can see in these pie charts here, you can see we sampled in the Mobile Delta, Perdido Bay, Perdido Key, Pensacola Bay, Escambia Bay, and Blackwater Bay. And the positives across each site are indicated with the pie charts. So the black shading indicates DNA positive. So black shading is where we found lionfish DNA. And then the gray is just where we didn't. So when we sampled, we did not get a positive at that particular site. But you can see we found lionfish at every single pie chart. Now, not every site. So all these black dots that are not having pie charts, we did not find lionfish eDNA. But we did find quite a bit of lionfish, eDNA well up into the bays and parts of the river system. So those dots that look like they're up on land, they're actually in the river system there. Okay, so what did we find? We detected lionfish in all months in all estuaries, and we had the highest lionfish DNA detection in July. So we had 17 of our 59 sites that were positive in July, as you can see over in the far graph there. And we also took water quality measurements. So surface salinity was an important measure for us. Now we just did surface sampling for this. So we didn't detect, we didn't collect benthic water or bottom water, and we just collect only surface water. And so we took surface salinities. And obviously, you know, those can range quite a bit in an estuary, and those can be different with the salt wedge that we have as well. So our surface salinities range from 1 to 22. But we had most of our detections at 10 PSU. So kind of an average salinity that we see, excuse me, across those bay sites. And then we also did some occupancy modeling, which is an important thing to think about with eDNA. And we wanted to analyze here whether the probability of detection differed based upon location, salinity, or season. And we used that eDNA occupancy package in R. And we used that to model probabilities of detection by fitting the Bayesian multi-scale occupancy model to our data. And basically what the result of this is, in this crazy table looking here, is that our data suggests that salinity plays a role in eDNA detectability of PCR replicates. So salinity, and we know that, does play a role in how DNA is preserved. And so that's an important thing to consider here. So the difference in salinity also likely played a role in our detectability. But nonetheless, we had a lot of detections of lionfish, which is stunning given that detectability with salinity. Okay, so for this story then, to wrap it up, we developed a mitochondrial marker to detect invasive lionfish in our aquatic samples. And using eDNA, lionfish were consistently, although depressingly, detected in estuaries in the northern Gulf of Mexico. And we had the highest detections in July and the lowest detections in January. Now, this is a little surprising to us. I mean, it's maybe not surprising based on what you might know about the fish, but in July, the water's hot. So there should be a lot of DNA degradation occurring and perhaps even lower detection. And we saw the opposite of that. So what that means is there's a lot of DNA in that system. Lionfish was detected in all seasons in Mobile Bay, which we think may suggest that there's residency in Mobile Bay. And it was detected at our farthest site, approximately 23 miles from the northern Gulf of Mexico. So as estuaries serve as a critical habitat for fish recruitment, lionfish present in estuaries could have serious ecological ramifications, and detection of lionfish DNA could have resulted from any life stage. So I just want to make that caveat. It's not necessarily a big adult lionfish, although it very well could be. It could also be eggs. It could be larvae. However, temporal sampling indicates that lionfish are likely in these estuaries. And I also want to note, we didn't go through a lot of the methods, but we did always sample at low tide so that we didn't have this influx of DNA, new DNA into the system at low tide. Okay, that was the first story. Now it's time for story 2. And so this story is about missing the mark. And so this paper was also published in conservation in another journal, but in Conservation Genetics. And this was work done with my former postdoc, Justine Whitaker, who's now a faculty member. And what we wanted to do was set out to look and just add data to the phylogeography database for lionfish. So, you know, there's data that are out there using D-loop, and this was from Johnson et al. In 2016. And we thought, famous last words, this will be easy. We'll collect a few lionfish and we'll add to the database for populations that are missing that we had access to in Southern Florida and so on. Well, as I said, famous last words. So that's our original objective, was to add to the data set. And we also thought, let's add another marker. So let's add CO1, which is in addition to the D-loop marker that was used. And we also decided, let's add a nuclear marker so we can just get a finer scale look into some of this evolutionary trends within lionfish. However, once we did some sequencing, we saw some major challenges. And what we saw was discordance between the D-loop and the CO1 marker, and that led us to do high throughput sequencing. So then we had to go to that next generation Illumina sequencing, and we had to do whole mitochondrial genomes to determine if this discordance was due to paternal linkage or pseudogenes. So basically, when we sequenced it, It didn't look like it should. It didn't look like a lionfish, like Pterois volitans or Pterois miles. In fact, it looked like both. And it looked like part of the gene was actually dead, which is what we call a pseudogene. It has a stop codon, so it wouldn't actually code for anything. Well, that can't happen in the mitochondria because those are essential and critical. So we knew something had to be going on. So that's when we looked into doing high throughput sequencing. So here's where we added samples from. We sampled in Pensacola, St. Petersburg, Jacksonville, Key Largo, in the Bahamas, and you can see how many different individuals we had from each site. We also added individuals from Trinidad and Tobago as well. And all these sites had not been sampled yet in the previous lionfish studies in terms of phylogenetics and population genetics. So as I said, we added the displacement loop. We added the cytochrome oxidase 1, that's a mitochondrial gene, and then the S7 nuclear gene. And so then after we did sort of conventional Sanger sequencing for that, we decided to add in that Illumina sequencing of the 7 fish that showed potential hybridization. One was from Pensacola, St. Petersburg, Key Largo, Jacksonville, Florida, and then a few individuals from Andros Island in the Bahamas. And then we also included 4 fish that did not exhibit hybridization, so we could use that as a comparison back to that. Okay, so what we did was add 163 new sequences for the D-loop marker to the 778 published sequences. And this is that combined network that you see up here. All those different colors represent places. So they're different locations. And you can see lionfish, by and large, there's several main haplotypes, what we call 6 or 7 main haplotypes. And most of the individuals fall into those main haplotypes, regardless of where they're from. So they could be from Pensacola or they could be from Trinidad and Tobago, but they have the same genetic makeup when we look at them from the D-loop marker. We also found a potentially novel haplotype from Jacksonville, Florida. And when we looked at the CO1 gene, that revealed a similar haplotype pattern. So these 2 markers revealed kind of the same story, if you will, in terms of the population structure for these fish. But those 7 that we looked at that appeared to have hybridization, in fact, we found that they did. So we found evidence of what we call pseudogenes using Sanger sequencing and then next generation sequencing. So in the top of this picture, we took the same individuals and we used this form called Sanger sequencing, which is conventional for 1 gene. And then we use Illumina sequencing to do the whole mitochondrial genome. And you can see that the bases that are highlighted in gray correspond to Pterois miles for the D-loop, where everything else corresponded to Pterois volitans. So for hybrids 44, 272, and 287, Sanger sequences corresponded to volitans, but sequences produced through next generation sequencing correspond to miles. Now the rest of the fish for those different genes look like volitans, but we have this miles integration that we're seeing into these fish. And so those gray squares represent those differences or those single nucleotide polymorphisms where we see that change. Okay, and so here's a look. You can see that kind of this, I'll show you the exact kind of hybridization that we see. So the bases highlighted in gray are the polymorphisms that correspond to P miles. And then this individual LF44 based on Sanger sequencing corresponds to miles in the first portion of the gene. So those first 3 polymorphisms, but then corresponds to volitans in the remainder in the genes. And that's what's in the box. This is weird. This is not unusual. And it's seen when we think about invasive species quite a bit because they're hybridizing. And that's exactly what we see here. And those of you that know lionfish do know that there are some meristic differences when we look at miles versus volitans, but they're generally hard to tell apart in the field. You just get this fish, and actually the individuals that we have correspond to volitans, so they look like volitans, not miles. Okay, so to wrap up that story then, we combined sequencing methods and that revealed pseudogenes from 3 invasive lionfish. And so that's basically genes that are incorporated from the nuclear genome into the mitochondrial genome, and that occurs with hybridization. And these pseudogenes can then present challenges for phylogenetic studies, which is why we titled this paper Missing the Mark or Marker, because choosing this marker, D-loop, that had been used across the board for lionfish, missed this hybridization and just showed it all as Pterois volitans. So in most cases, pseudoargenes are identifiable by the presence of stop codons. So what that means is this codon that when you see this sequence, you know it's not real because this gene won't be amplified. But in D-loop, this is a non-functional gene that possesses stop codons. So this was probably missed in this research before. So previous lionfish studies probably led to erroneous phylogenetic conclusions, or maybe these sequences were discarded because they did have those stop codons, but they were actually valuable information and tell us about hybridization and potentially paternal linkage between two species of invasive lionfish. So multiple markers is key when we're thinking about robust studies and conservation management. And I think I have time to tell you one last story where okay. So this is about characterizing the diet of the least tern using DNA barcoding. So understanding diet is an important component for an ecological role of an organism. And when we think about conventional methods, we use direct observation, we look at stomach contents, and we also look for drop fish near colonies, particularly when we look at birds. And so what we did was basically collect bird poop and then extract DNA from it and then sequence the DNA from that. And that's a non-invasive, accurate, and then also potentially cost-effective solution to look at diet. So we did this, and I never thought I'd tell this story in a lionfish workshop, but what we found were some lionfish, and so that's why I'm telling you this story. So what we did was identify multiple species with metabarcoding. So we take a sample, and then we can use generalist primers and what we call a universal primer, and then try to amplify lots of different types of fish species for that. So it's a more for less, basically. And just to give you an idea of more for less, this was sort of a revolutionary paper that was put out in 2017. After 14 years of visual survey used to detect the same numbers, after 14 years of a visual survey, they used eDNA, and in 6 hours, they had the same species that they found in 14 years. So I always say that, like, in the 5 years I did my PhD, you could now do it in a day, but that's what happens, right? With molecular methods sometimes. So we wanted to use DNA metabarcoding to examine the diet of the least tern, and we wanted to compare priatoms of individuals from breeding colonies over a 3-year period to better understand the inshore food web. And so we also compared those to drop fish data. So like you see this bird here holding a fish, they often drop those fish. And so we compared that. And then we also compared adults to chicks. And I'm not going to tell you about all those data today because that's not relevant for lionfish. But if you want to talk about that at the break, I'd be happy to. Okay, so here's where we sampled on the northwest coast of the Gulf of Mexico, but this was more over in Mississippi. And we collected in 2017, 2018, and 2019. Unfortunately, we didn't have the same sites because birds don't always use the same sites. They go to different places, and that's what you can see represented here. And then we classified them into different zones. So zone 1, 2, and 3. And so what you can see here are the fish prey items by zone. And what I really want to point out to you is the second bullet point here. So yes, we found a lot of Anchoa mitchellii, which is a really common fish that we see with least terns. And we also found Fundulus grandis and Medina, but we also found a lot of lionfish. And they were really present in zones 1 and zone 2. And those were the first two kind of bigger boxes that we showed you. Zone 3 was more of like freshwater habitat, like up on a colony there. And so this surprised us quite a bit. When we got these data back, we were like, lionfish, this has to be contamination. They were like, no, we put controls at every single step. And then you can see the lionfish are represented by green. So it's like the third most common species that we see. And you can see that's different across the zones. And so that temporal sampling across the zones, and then also across the year, oh, there's the lionfish for us in green. And then the other species there in blue. We see differences in these different sites and differences across the years. And so across the years, I'll just go to lionfish for you. There we go. And across the years, what's interesting is that we had a lot more lionfish in 2017, fewer lionfish in 2018, and even less in 2019. And so you can see those sort of numbers of detections across the board there. And this is correlated with some data that we also see in the Gulf of Mexico. Let's skip over the other fish since we're kind of running low on time. So just to wrap that up, we had a total of 21 different fish taxa from 83 different fecal samples, and lionfish is a new record for the least tern diet. It could be eggs, it could be larval, and it obviously is going to be juvenile fish. It's not going to be a big adult lionfish, but we don't think this is due to secondary predation. So we don't think this is Anchoa mitchellii eating lionfish larvae or eggs and then having that DNA inside of them, inside of the lionfish. And the reason for that is because the amount of detection that we have, and then also the temporal detection, although it could very well be secondary predation. They were detected in all fecal samples from all 3 zones, but mostly in zones 1 and 2. And lionfish detection decreased over time, which may be correlated with declines in larval lionfish recruitment. And the diet shifts may correlate with prey abundance, which is what we do see with the other species as well. So we see a change in that fluctuation with prey. This work was done with both postdocs in my lab and then Abby Dara and Molly Folkerts at Audubon in Mississippi. And of course, there's lots of people to thank for all of this work in terms of funding, University of West Florida and NOAA. And then, of course, my contact info if you want to reach out. And maybe we have time for a question or two in a little bit. Okay, thanks so much.
Any questions?
Matthew.
The bird species that you were looking at, the fecal samples for finding detection of lionfish DNA.
Yeah.
What are the typical predation habits of that? Where is it eating? Where are we seeing it? Is it snooping from surface water?
Yeah, surface water. So the least tern is the bird that you sometimes see hovering over the water, and then they make this dive down into the water, and they get usually a small-bodied silverfish, kind of tiny little fish, and then they feed it. They sort of display it as a breeding behavior, and then they also feed it to their chicks, and they eat it themselves. So these are usually small-bodied fishes. What's interesting about that is when we looked at the chicks compared to the adults, drop data show us that the chicks eat only the skinny body or the narrow-bodied fishes, but drop data shows us that the adults bring lots of different size fish, but the chicks only eat the small ones, which makes sense because chicks are small compared to the adults. But so we think that we didn't see any dropfish, lionfish data, but we do see it in their diet. Mm-hmm.
I hate my.
It's fine.
Going back to the environmental DNA, I'm assuming that if I thought of this, you surely thought of it. So what is it in your research that you know that shows that this water didn't come into contact with lionfish DNA somewhere offshore and get blown in, or?
Yeah, that is definitely a possibility. We sampled at low tide. And the way that, an estuary works, particular here, is like one high, one low in a 22 or 23-hour period a day. So we always sampled at low tide when the tide had already gone out. So after slack tide and the water's moving out. So the water's in theory coming out of the river, right? And into the estuary. Now DNA can persist and it can stay in that system for maybe 2 or 3 days at the longest points in terms of time. But our temporal and repeated sampling gives us that good indication that it's not just coming in from the Gulf of Mexico. It's a good question. We were worried about that, and you want to think about that for sure. Yeah.
Thanks.
Mm-hmm.
Any other questions?
Thanks.
Thank you. All right, so we're going to move right on along. Keith Milley and Robert Turpin are up next. They're going to be talking about monitoring PCB concentrations of lionfish that were collected from the Oriskany. So, gentlemen.
Yeah, thank you, Thomas. This is an update on the work that many of you have been helping us volunteer with. Dozens of folks have helped catch the fish that we've been analyzing from the Oriskany. So we're very pleased to provide an update on this. And also, this is a boiled down version of a presentation we've given many times before. And to make it more lionfish specific, we kind of try to pare it down a little bit. As a little bit of history, the USS Oriskany measured originally 900 feet long, beam of 128 feet, and it was launched in October of 1945, decommissioned in 1976. Reef planning started in 2004. The EPA issued the risk-based PCB permit to the Navy in February 2006. and was deployed as an artificial reef off of Pensacola in May 2006. So Robert and I wanted to kind of give a little bit of history about the background on the EPA risk-based PCB permit. This was the Navy applied for and received this TSCA permit from the EPA specifically to allow materials on the Oriskany that contain PCBs to remain on the Oriskany. This was a cost-saving measure. They spent, about half of their budget was spent to do a lot of human health toxicological analyses to make sure that it would not pose a human health and safety hazard. And that permit was issued on February 2006, following extensive science consultation meetings, public workshops that we had at the Civic Center in Pensacola and such. And also, Robert reminded me that the EPA guidelines for best management practices for sinking of artificial reefs that was published in May 2006 was an outcome of the planning and the work that was done in preparing the Oriskany. So the human health reasons for monitoring polychlorinated biphenyls on the Oriskany reefs, they are persistent organic pollutants. They have been banned in the United States since 1979. They're capable of bioaccumulating, a probable human carcinogen. And the concern here was exposures to recreational fishers by the fish that they caught. So issuance of that permit, and here's a summary of the PCBs that were left on board the Oriskany, about 723 pounds of solid PCBs. Most of that was represented in the cable insulation. And then there were some in the paint and bulkhead insulation, rubber gaskets and vent. All lubricants, hydraulic fluids and such that contained PCBs at the time were removed from the Oriskany. So issuance of that permit by the EPA to the Navy it included a monitoring plan that after the sinking, the state of Florida, FWC, and Escambia County were responsible for complying with. And that required us to monitor these fish. And if the PCB value of 20 parts per billion was exceeded in 15 or more legal size recreational fish, then we would continue sampling. And if it was lower than 20 parts per million, then we would be able to discontinue sampling. Okay. So the criteria for the Tier 1 monitoring plan was that we would sample 30 recreationally targeted legal size fish annually. We collected skin-on fillets, which is a little more conservative in that you're including the lipids in those skin as well. And we analyzed all 209 PCB congeners. Again, minimum of 15, I'll mention this a few times through the presentation because this is key. Minimum of 15 specimens of a single species was necessary to compute the average PCB value. Our target species were initially red and vermilion snapper. And again, if the mean PCB for a given species exceeded that level of 20 parts per billion, we would have to consult with EPA and initiate Tier 2 monitoring. So a little bit of background on the location. It's 23 nautical miles southeast of Pensacola Pass and sunk on May 17th, 2006. It's located at an ambient depth of 212 feet and has about 80 feet of navigational clearance. And it's about, about 150 feet to the flight deck at this time. So here's some images from the first day after deployment. We had spectacular visibility despite the 6 to 8 foot seas that day. Some of these pictures continue to roam the internet and some of our publications. Those of you that might have picked up our artificial reef brochure that we just published this week had that photo on the cover. That is Robert Turpin there on the left. And, we started doing our fish census pretty early on. Here's Tim McIntosh of Miami-Dade County, actually, at the time, doing some counts. And it wasn't too long before this became popular with scuba divers. In 2007, Artemisia, now Reef Smart, did one of their first dive maps on the Oriskany. And this illustrates the typical dive that an average recreational diver might have. And this, diving this control tower would all be less than 130 feet. So, by 2.4 years after deployment, we started to see some fish assemblages. Here you see a high density of vermilion snapper and other fish species. There were some structural changes that occurred. The most prominent one was in 2009 due to Tropical Storm Ida, which removed some of those removable panels that were on the side of the smokestack, allowing for some swim-throughs. And then by 10 years after deployment, we started to notice that the deck started to open up a little bit more, as rusting was happening, and started to see that. Here we are 16.4 years. And again, this is the top of the smokestack, comparing it to that early photo, you can see how that started to open up. And again, the deck is getting a little bit more open up. Our most recent dive was September 2023, 17.3 years. And, I zoomed in a little bit and you could see some of those cables with the insulation that presumably has some of the PCBs that were left on board from the flight deck and below. I should mention that all wiring was removed from the flight deck and above. So that control tower has no wiring in it. It was intended to be made more diver safe. And in doing so, presumably PCB free as well. But the flight deck and below, you will find a lot of these cables still in place. The results of our fish census, and again, this isn't really intended to read those specific species, but early on, and we quickly identified 53 species. The targeted species are those that are highlighted, and so the ones that we actually analyzed are in yellow. So we captured all and analyzed the ones not only that we targeted, but some additional ones. The recreationally targeted fish are indicated with those red dots. The Almaco jack and greater amberjack were two that we did not analyze because those are species that are more transient. They spend their life cycle mostly above the structure, not really affiliated with the structure of some of these other reef fish species. So let me get to the collection mechanisms, because this is part of the story that brings us to lionfish eventually. We started the first sampling event, first 1 or 2 sampling events was using chevron traps, and we hired a commercial boat, and we quickly discovered that it is not, putting fish traps down next to a large structure like the Oriskany is very challenging, especially when you're dealing with currents and seas and such. So we lost at least a couple of traps, had to send divers down to retrieve it. It weren't giving us the numbers that we needed. But while we were there, they had a bandit rig, and hook and line turned out to be an effective way to catch the fish that we needed. Again, we just needed to get the fish onto the boat to send to the lab to process these fish. We had to do it as efficiently as we could. So that eventually brought us to realize that we could effectively catch these fish using hook and line. And many of you, or some of you in this room, have helped us over the years with our annual volunteer Oriskany hook and line fish sampling event. And that helped us catch the 30 recreational fish that we needed. And sometimes, in the early years, we were catching those fish that we needed in less than an hour or two. Some of those early years, we even stayed an extra hour and tagged 200 and released over 200 red snapper. And that helped kind of towards a little bit of a tagging study as well. However, it became increasingly more difficult to use hook and line to catch the 30 recreationally targeted fish that we needed to. So we actually implemented divers, and they targeted some native reef fish species, the grouper and the great triggerfish and such. And that was effective, but there was a lot of effort that went into that, and a lot of uncertainty. You don't know, and it was also heavily based on the skill level of the spear fishermen. Here's our good friend Brian Clark, who many of you had known with his catch on that day. And then it eventually transitioned us to lionfish. Now, remember, lionfish only was first observed in the northern Gulf of Mexico in 2010. You might be able to see it on our little map over there. So, by 2013, we started to see lionfish more regularly, and hear the dive masters, collecting it regularly from the control tower. we eventually, you know, to collect the 30, to get to the point of collecting the 30 lionfish that we needed, you know, more than 15 times 2, it really required technical divers going below the flight deck. And, you know, we found that we were able to successfully the past couple of years collect the 30 fish that we needed in one long, you know, decompression technical dive using some helium and rebreather divers. Matt is Hardman's in the audience today. I appreciate the work that Matt had done. You know, it became an effective tool for us. When we look at the species composition, so this is the other part of the story. The first part is the collection methods, second part of the species composition. Initially through round 10, red snapper dominated our catch. Granted, this was all hook and line, vermilion followed by vermilion snapper. So this is why I mentioned vermilion snapper and red snapper were our initial target fish species, because we could reliably catch, you know, the 15 to 20 fish on it in one trip. So here are the results of some of the work, and I'm just gonna kind of gloss over these so we can get quickly to the lionfish results. For vermilion snapper, you see in the, you know, in the first 12 years, we never exceeded that 20 parts per billion. And again, 20,000 picograms per gram is 20 parts per billion. So that green line represents that threshold that the EPA established to determine whether or not we would continue sampling. So vermilion never exceeded that. So as a result of this, we decided that you know, our money would be better spent on other fish species, you know. So let's go to red snapper, because that's where we have the most amount of data. But before I zoom in on that 20 parts per billion, Robert and I wanted to share with you the EPA short-term acute consumption limit, and that's 2 parts per billion. And you see it's logarithmically above the level. This is the level that it would be actually unsafe to eat one fish, right? The other levels, the Florida Department of Health, 50 parts per billion is the level at which they might consider a consumption guideline, depending on the number of fish that are collected. So these are the results for red snapper, the species that we have the longest term data set for. And I mentioned early on in issuance of that EPA disposal permit, there were a lot of toxicological studies that were done, lab analysis, short-term leach estimates, and they actually anticipated that within the first 2 years, there would be an initial peak detection of PCBs as they leach from the PCBs that were left on the Oriskany. And they anticipated that would drop down, and the laboratory studies did not. Where they missed the mark was on the amplitude of that peak. So the shape of this actually did reflect what the laboratory study showed, but where they missed it was we actually found that we were catching fish above that 20 parts per billion. However, by year 4, it dropped down below. But the EPA, because we were still kind of straddling that line, the EPA required that we continue our annual monitoring. And so this is where the fish assemblage story comes into play too. Over time, the fish assemblage changed on the Oriskany. No longer were we able to catch the 30 red snapper that we were in the early years, and it became increasingly more difficult. And I'm talking about to the point where we would have to do 2, 8-10-hour trips of fishing, 15 poles in the water, and only come home with 4 to 3 fish. There were some samples where it was even 0. And so that's where we kind of said, well, we need to start thinking about different collection methods. Again, we just needed to catch the fish. It didn't, and we were unable to do that. We could not spend 30 days out there to try to catch 30 red snapper fish. So as a reminder, we started with the fish traps. We switched to hook and line, and then we added that spearfishing with Brian Clark and others to help supplement. And that kind of got our numbers up there. we never did really get a solid, because now we're catching all different species. We never were able to get that 15 fish of any one species that we were needing, especially once we dropped vermilion snapper, you know. And so here's the results. So in the process of this, then we started to look at some of the other fish species, and that brought us to, when we were just analyzing red snapper, we had a nice clean curve. That was our focus. But then we started to pick up all these other species and look, whoa, wait a minute. Now these species are more than 20 parts per billion. However, we weren't catching the 15 fish that we needed to make any determination if we needed to continue sampling or not. So it was very struggling, you know. However, you know, by 2013 or 14, lionfish really started to kind of, you know, become resident on the Oriskany. And so that's when we added the lionfish sampling over there at the end. Okay, so that would have been, so here's the result, though. This is the conclusion of the of this long background story to get to where we need to be. You know, these are the results of our lionfish analysis. And I should mention that, you know, during year 11 and 12, while we were down there spearfishing and doing our other fish samples for those other native fish species, we did bring in some lionfish too to check on them. And most of those were all less than 130-foot depths. And so we found that, wow, we were detecting it in lionfish. And we were hoping to drive the project down to, you know, wind it down. And, you know, the EPA really wanted us to continue sampling. We determined, well, okay, let's focus exclusively on lionfish. And so that's what we did the past 2 years. We collected 32 fish 2 years ago and 30 fish this past year. The average just this past year was 15 parts per billion. So we're now below that threshold. I think the jury's still out to see if that will, you know, but will drop. So I think our goal now is to continue sampling in 2024 and see how that brings us, and then reconsult with the EPA to see, you know, how close we are to bringing this to a close. So some concluding interpretations here, and Robert, chime in, you know, as needed, and then Robert's going to jump in at the final slide here. No vermilion snapper samples exceeded that EPA threshold. After 3.5 years, red snapper PCB levels were below the EPA threshold, but then they were elevated, and then eventually they dropped down on the few fish that we were catching. The remaining analyzed samples were too few, with too great PCB variability for the Department of Health to make a species by species determination. And by year 8, red snapper catch rates have been low. You know, as you saw, there were very, very, very low numbers since then. So the highest PCB levels were initially red snapper, scamp, bank sea bass, graysby, red porgy, white bone porgy. And now amongst all this, I should mention that in 2017, we did catch more than 7 bank sea bass and scamp grouper for the Department of Health to make a determination. Now that number 7, that magic 7, was the number for the Department of Health. It's again not affiliated with the 15 that the EPA mandated us to analyze in accordance with the EPA disposal permit. Overall, in general, the catch rates have dramatically reduced. The fish assemblages have changed over time, and we've exclusively focused on lionfish since 2022. So all this kind of brings us to the safety, seafood safety considerations, and I'll pass it to Robert to kind of fill in any gaps that I might have left, and bring us back to the whole reason why this sampling is required.
Thanks. And during the Navy and the EPA discussions and deliberations about the implications of PCBs on the Oriskany, we were present in the room, and it was very interesting to hear the Navy had consultants that took samples of parent materials, the solid phase wiring and bulkhead insulation, paint, and they did leachate studies. And it was very interesting to hear how the modeling that occurred that allowed the Navy to convince EPA to issue the permit that said there will be no long-term adverse human health effects. And but once the ship was deployed as an artificial reef, that ended the Navy's role and began the Escambia County and FWC, State of Florida Department of Health. It began our official role to then take up the sampling and analysis. And every year we've through funding, through grants, from FWC, we've been able to go out and collect all the data that Keith has shown you. As someone who lives in Escambia County and who eats seafood regularly, part of my mission at Escambia County is to consider the effects on me and my family and my community. So we actually were very keen on, what we saw above that 20 parts per billion and the 50 parts per billion, and how that would influence our role in Escambia County. And what it resulted in was we started conducting seafood safety symposia. And at the first one, there were more of us in the room that were staff than we had. I think we had 2 attendees at our first. We still continue to do these annually, because we want the public to know that not only are there fish advisories for the Oriskany, but also for other fish species, for things like mercury. So part of our message of seafood safety was derived from our experience at the Oriskany, and then when we started looking at mercury and other constituents in the diet of our seafood consumers, it became evident for us that we wanted to continue to outreach, that there are fish advisories on almost every saltwater fish. And those consumption advisories are important for all of us. In doing so, we decided that we would actually start collecting data on fish consumption surveys in Escambia County from our around Escambia County. We in Escambia County have a handful of recreational fishing tournaments, including lionfish tournaments. So Connor is here. He's my intern coordinator, and in conjunction with these fishing rodeos and other outreach and education events, we've developed a seafood consumption survey. So we ask people, have you fished in the last year? Have you fished at the Oriskany? So we had from 2016 to 2023, we've conducted 647 surveys of the fishing public. Of those, 102 people responded that they had caught and consumed fish from the Oriskany. The number one consumed species was vermilion snapper, and there were 641 of those. There were 355 red snapper that were caught and consumed. The next was 135 triggerfish, but lionfish shows up as ranked number 36. So it's important for us to continue to give this message. And lionfish, I think, are a really important part of our sampling, and especially for this human health, because as far as we know, lionfish are very they have a high reef affinity, especially at the Oriskany. They are a fish that has no closed season. There's no minimum size limit. And we're also promoting lionfish to be removed by divers and anglers and consumed. We're promoting their consumption. So I think we as in our positions with the Escambia County and FWC and throughout the management, we should be communicating all of our data with the Department of Health, but also back into our community. So that's why we're really excited to present these results. We do this annually at Escambia County's Marine Advisory Committee. We do a seafood safety consumption. We will be having one, I believe this year we're going to do it in April. And we will be collecting lionfish again this year. So until the EPA says we are no longer obligated to monitor the Oriskany. We're going to follow their original guidance, and we'll continue also to do our outreach and education through these seafood consumption surveys. So it's really an important way for us to take the information that we are collecting and provide that to the public. So with that, I think I'll turn it over to Keith and we can answer any questions.
Yeah. Let's, for the benefit of the people online, let's have stand close to her so we can hear it into the mic.
Okay, I'm being a question hog today. Two questions, actually. The first one's pretty short. Do you have controls, or is there presence absence that says that the Oriskany is actually what's causing the higher levels? And then secondly, some of the species that showed a spike in levels around 10, 11, 12, does that correlate with when the deck started to disintegrate and the wires were more exposed, there were more fish able to get down to that? Or was it, did those PCBs eventually all leach out, or are they getting buried with sediment and colonization by other critters?
So I should first maybe preface all this by describing the EPA's Tier 1 monitoring, which is the category that we're in, the lowest level of monitoring. And it's not intended to answer some of these questions that you're asking, right? Merely Tier 1 is merely, are 15 of any one species exceeding the mean exceeding 20 parts per billion. And where that 20 parts per billion came from was kind of out of a hat too, because the Florida Department of Health uses 50 parts per billion, So, we did, however, allocate some additional funding to go to a nearby reef site. We're not calling it control. The EPA says in order to have a control, we would have had to have the Oriskany and her sister ship, one with the wires and one without the wires. Only then would it actually be a control. So we went nearby to the Chevron Reef, and we analyzed fish from there. And most of those were non-detects. I mean, maybe like 1 part per billion. The nearest PCB contamination site was located way up in Pensacola Bay, the Monsanto site, 85 nautical miles away. And, so these are the slides that I removed from this presentation. We did all kinds of things. Yeah. And again, I'm glad to share those. In fact, if you look at the poster that I had up yesterday, I had a little bit of that stuff in there. We looked at the PCB levels and the size of the fish, which can be, and determination help point to, is this a point source, or is the PCBs kind of ubiquitous, in the environment? And we found that, the highest levels are actually in the smaller fish, not the larger fish that are spending their time out other parts of the Gulf of Mexico. We also did look at we analyzed all 209 congeners, and the gaskets, well, actually it didn't work out because it was only the gaskets that had a unique congeners. This goes back to your organic chemistry classes and all those, organic chlorines that are around there. You can actually, it creates kind of like a fingerprint of the chemical. There is some biodegradation that happens, and then with bioaccumulation, it kind of blurs it. So, we looked at the whole spectrum of all those 209s, and unfortunately, we weren't able to kind of tie that fingerprint back to the Oriskany specifically. So I think anecdotally, the evidence points to, it seems like it's coming from the Oriskany, but at the Tier 1 level monitoring, it's not intended to dive into the details and look into the trophic levels, because the next question is, well, what are the PCB levels of the prey that are being consumed by these fish that we're detecting as well? That is beyond the scope of the Tier 1 monitoring. Human health is the primary objective, right? Yeah. So long-winded answer. That's basically a lot of the background stuff that I pulled out of here to try to just talk specifically about lionfish.
So anybody that wants to come help us this year, collect lionfish, see Keith and or me, and we will sign you up and let you know when we're headed out there.
I should mention though, lionfish, as many of you who have been on the Oriskany, are not that common on the control tower and above because you guys are doing a great job at removing them. So this trip, unfortunately, the prerequisite would be, trimix or rebreather, some deep technical divers that can get for the flight deck and below, because that's where the densities of fish are that need to be harvested. Yeah.
They know you're coming.
Yes. Thank you.
We're ready to get started on the next session. We're going to be doing a commercial harvest update. We're kind of changing that a little bit where me and Rick had gone and surveyed some markets and some seafood restaurants in our area just to see their response to.
Here's some bring in there. We don't have to do that. See if that's better.
Test 1 2 3 test. Just to kind of see what the attitude was in Pensacola when it comes to getting lionfish in the restaurants and markets. What can we do to get it on a plate? My name is Thomas Sturvis. I'm the Sea Grant agent for Santa Rosa County. This is Rick O'Connor. You probably know him. He's the Sea Grant agent for Escambia County. Together, we're trying to figure out how to get lionfish on plates. We seem to have running into an issue in Pensacola of getting them on plates. And when we talk to people that are here in Destin, they tend to be like, well, we really don't have an issue getting it on the plate. The problem is the reliability. So I'm going to give it over to Rick. He's going to be doing the white slides and I will be doing the blue slides.
Yeah, how this all started, actually, we were contacted a few months ago by some of the providers of lionfish and wanting to know why some of the seafood markets and restaurants were not interested in what we could do. And, I right away texted one of my seafood contacts and I said, are you guys interested in lionfish? And they basically said, no. I won't say everything was there. They basically said, no, they weren't interested. So that drew up curiosity. Obviously it was no, and there was a reason there was no. We were trying to figure out what that problem was, what those barriers were. And then maybe we could work with them. And we weren't going to try to make them buy lionfish. That wasn't really the objective. It was just to find out why it was that they were very quick on the draw to say no. And then as we were having that discussion a little bit later, we heard from the Destin, Panama City area, it's not as hard. So I said, okay, that would be a good a talk topic for this. Find out what those barriers are in the Pensacola markets and how, if we got those barriers here, I'm sorry, if we're overcovering those barriers here, find out what those bullet points would be, and then we could go back and share that. And just show them this is how you get past this. This is how other folks are getting past this. So you can see here we got on the slide the chalkboard entry. If we can get it, we will cook it. Once it was a couple of on the menus, and it's not anymore. Featured only in one restaurant, as you can see over here in the Pensacola area. The new lionfish tournament is on its way. We'll slide down here. Well, why don't you provide, what do local markets and restaurants think? Why don't you provide the lionfish? And I will go ahead and you'll see a couple up here and you jump in because you talked to even more than I did. The first thing I heard, and we had a gut feeling about this, was the size and the yield. That was the first thing we heard. And you agree with that, with the other ones you talked about? That ended up rising as the #1 barrier. The contact I talked to, they said, look, when I get there at the market first thing in the morning, if I've got to lay fillets out and I got to have things on ice and ready to go by opening time at 8 o'clock, and I put, let's say, 4 or 5 pounds of fillets out. I've got a cooler here with grouper or snapper. And then I've got this cooler over here of a bunch of little tiny lionfish, and I've got to clean that. He goes, that's a problem for us. And we got to figure out how to get past that. So that was the big thing. And then again, the percent yield, which again, as you can see here, 20 to 25% is what they're getting. So that was barrier #1. That was absolutely barrier #1. And one of the other things you'll see over here in the second, and again, you pipe in because the order may have differed from the folks that you talked to. There was still concern about the venomous spines. We've been talking about that since 2010, 2011, but there were still issues and concerns about that. And then, as I brought it up to them, Well, you know, if we showed you how to clean and fillet these things and it's not as bad as you may think that it is, would you be willing to do this? Are you interested in learning that? And the answer was yes, they were. But at the same time, once we go through, and then again, we'll talk about some of these other barriers that they've run into, you know, the bottom line is even if we hold a workshop for the folks over there, show them how to do this, if we still have that first barrier of yield and size, we still get, that seems to be the number one barrier. So again, as we go through this, we'll have some talking points, but with some of the time, we hope to hear back. I don't know if anybody here today is with a restaurant or a seafood market and is one of those who have solved these problems. We would like to hear that, make those points on note. And then when we turn around, we do plan to talk to the markets, all of them. We talked to one of our last questions. We'll go through our last questions. Are you interested in education? All of them said yes, all of them. So yeah, there are some issues, there's some concerns, but they're willing to listen, they're willing to learn, and we'll see where that goes. You'll see a couple other things, you know, a dime holding up $1. You heard that, I heard that. Little or no demand. I did ask my contacts again, all right, since it started, how many times have people come in and asked about lionfish? Because we'll hear later from the education outreach, we are doing the education outreach on that. She said in the last 10 years, one person has asked. So that was all that it says. So we don't know. But I do believe, and I think all of you sitting here believe too, especially if you've been involved with education outreach, one of the questions I always hear when I'm in an event, people will come to my table, where can I get it? You know, I want to go buy this. So I don't think that's going to end up being a problem. Cost is too high for too little yield. That's another issue. Everybody's been hearing that over about 10 years. Markets can clean more fish in time than it takes to clean just one lionfish. So we kind of talked a little bit about that. All right, blue slide.
So like you said, when I was talking to a lot of these markets and the restaurants, it was kind of cool because they're all saying the same thing. So it's kind of a nice consensus that everybody had that, you know, another big risk was the venomous spines. It wasn't just for the person that was filleting the fish. It was actually for the customer, say that they went to a restaurant and they ate a whole lionfish that was fried. They were worried about the spines. You know, we were giving them options of, well, of course you can cut off the spines before you fry them and cook them and serve them. And like I said, the answer for the venomous spines was pretty much given by every single restaurant and market, which I think is kind of crazy to think about because I feel like we've gotten to the point now to where we know how to handle lionfish and clean them. And they also mentioned that the cleaners don't like touching the lionfish, which I find that a little odd because they'll touch every other fish that they get. And it's mainly because of the spikes. And so we were wondering, you know, how can we go about educating the markets and the cleaners that are at the markets on how to properly handle lionfish? We all know that there's only, about 16 venomous spines, 15 venomous spines on the lionfish. So if you cut them off and get them done, it should be easier to clean. And like I said, one restaurant mentioned customers potentially being spined when cooking and serving the lionfish whole. Most of them all mentioned that. And we also, you know, what kind of advancements would you like to see when it comes to getting lionfish in your door? The number one thing was consistent sizes. Everybody said that they wanted a consistent size. There was one surveyed chef that I had talked to that actually served lionfish at a time. And he was telling me the worst thing for him is that he would have to serve a plate for one person that had 6 or 7 fillets of lionfish on there to make it seem like it was worth the value of the lionfish. He was telling me that he was selling his lionfish plates for $45 for the plate, and that was pretty much not getting any profit whatsoever off of it. He was just trying to get it out there, get it on a plate so people could eat. One surveyed market said that a way for them to get it on their door, to get through the door, is to have them pre-cleaned, processed, and in a bag. And in a great world, yes, that would be amazing. But if you're talking about lowering price, that's the opposite. That's all that's going to do is increase your prices. Another one, they said lower price, markets and restaurants. Consistency with supply is key, and we understand that that's kind of hard to do, especially since lionfish isn't really a trap or a hook and line fish. It's mainly just to dive down, spear it, and get it. They really were into the education for consumers and the restaurant owners. I had talked to one restaurant that said, yes, if you can get me lionfish, I'd love to put it on there, but we have to get it past my owner first, who doesn't know much about the lionfish. And so maybe if you got the owner on board, he'd be more gung-ho about putting lionfish on his menu. And then also we were thinking about doing cooking demonstrations and recipes. There was a restaurant that I had approached that said he would love to go to a seminar on learning how to actually handle the lionfish, how to properly cook it, and how to have different recipes for consumers. So not just a whole fried lionfish, doing something like a pan-seared lionfish. One of the restaurants that I had surveyed said that they would love to do lionfish sashimi, which I thought was a really good idea for them. But the biggest thing is that they really wanted a lot more education on it, especially when it comes to the consumers and to the owners. And I think that if we provided that with them, it might open up the doors a little bit more to lionfish being at a market or a restaurant. I'm sorry about the slides being like that. It didn't look like that when I submitted it, but covering up some letters.
Yeah, I will echo one of those. One of my seafood market contacts, they said, yeah, if you can have Cisco truck drive up with a bag full of lionfish fillets, we'll market the heck out of them. You know, so that was kind of one of the things they were looking for. And I don't know if that's going to happen. Would you be interested in learning more about lionfish, such as the cleaning, processing, and benefits? And as you can see here, every market said yes, and every restaurant said yes. And as I talked to this, and again, I don't know if they were trying to make us feel good. You know, I don't, you know, because a lot of times I'll hear that. Do you want to learn more? Oh yeah, I do. You know, like whatever, dude, say what you got to say. I already know what I'm going to do. But I really do feel they do. They want to learn more. They're interested in it. I believe you said some of your restaurants had it on the menu. It's not now, and they'd like to bring it back. Is that yes? So they would love to bring this thing back. So they're all open. And as you can see, some of the comments here, which are kind of hard to read, if I could provide lionfish and knew it would be a safe product for cleaners and customers, count me in. If I could learn more how to be quick and better at processing the lionfish, they would love to learn how to do that. I had lionfish on menu once. There's that comment. We heard that one. And now I'd like to get it as a mainstay. That is also a problem. And again, when I'm done, I've got notes here in case some of you have some feedback for us. Early on, I was contacted by a chef from Charleston, South Carolina. This was back in the heyday when densities were still relatively high around here compared to what Will was telling us this morning. And he goes, oh, I heard you're in Pensacola and you guys have got lionfish all over the place. I said, yeah. He goes, man, we'd love to buy this stuff. I said, okay, great. We got people want to sell it. How much do you? He goes, I need 500 pounds a week. Okay. You know, so again, you come back to that, you know, they want it on the menu, but they want a lot. I mean, to put it on the menu, and I get that. You don't want someone to walk in, open up the menu, see lionfish and say, hey, sorry. I happen to go into a restaurant. Some of you in Pensacola might remember Cypress, which is down there. You know, they served it one night. I walked in with some friends and it was on the chalkboard and they said, oh, lionfish. And they go, you're going to eat it? Like, yeah, I have to, right? I've been saying this for 10 years. I don't have a choice. I lionfish. So it was $33 is what they asked. But I found out that was only 3. They only had 3 that night. I got one of the 3 lionfish. So that's still an issue or problem there. To get past that. But again, they know what their barriers are. We're learning more. It does sound like, again, a big problem is the amount of cleaning they have to do, and the processing part seems to be barrier number 1. The spines are still an issue out there. And as you saw from this, there's some other problems, but they are willing to learn. Nice. Yeah. And we're open. It's like, wow, it kind of fades and go away. That's nice. All right, open discussion and questions for the audience. This is kind of, we wanted to get some feedback from you guys. And you'll see the first question there is how could we get lionfish to a market or a restaurant? In the area here, Destin, Panama City, and then again, if you don't have feedback, we would love to hear from you a way how they deal with this particular problem. I mean, let me ask, is anybody here affiliated with a seafood restaurant or seafood market or affiliated with selling it to these folks and know how you're getting the product to them and at what frequency? No, not really.
Problem for the divers and the guys out there doing it. The juicing worth the squeeze. I mean, going out there and doing it, it's dangerous. You know, and when we go, I can pull a couple 100 pounds of lionfish in a day. I mean, I've got the guys that'll come do it, but I got to pay them to do it. I got to pay for the boat to do it. And then, perfect world, we're not going to pull in 200, we're not going to be able to have consistent numbers. I'm good, but as any boat captain can tell you, some days we just don't hit the sites. And then the other side of it too is just the liability behind it, you know, I mean, throwing divers in the water and when now we're out at 120 or 130 feet, and we're, you know, we're cycling eco, you know, now I mean, there's so many dive components to it that just make it, it's a dangerous sport. And the last offer that we had, they all had to be a certain size, and I don't remember what it was, but it was good, you know, I think he wanted better than 10 inches on every one of them. They had to be face shots, and then I think he wanted to offer me, it was $5 or $6 a pound, and I thought, you know, it's just, when you start doing the math, it's not really worth it.
Yeah.
You know, where I can just run dive charters and run lionfish charters, and, you know, my liability is signed off on a liability waiver for those guys to do that. So, you know, if the money was there for it, I mean, I'd love going out, and I've got groups of people that would love to go do it, but not for the money that they're offering. So, you know, and I get it, there's a balance on that. It's just when he offered, I kind of emailed him back and was very respectful and said, I'm not interested in, you know, not for the number of requirements. Because then if I didn't meet all the requirements on every fish, you know, now my price got cut in half. Well, and then there's the problem of time. So now I got to take 2 or whatever, we'll just say 200 pounds of lionfish, and now he's going to cut my price in half. And I'm at the mercy of the guy. I'm not going to sit there and watch every single one. You know, I mean, so, you know, there's that part of it too. So at the end of the day, for me, you know, somebody who's out lionfish hunting multiple times a week, it's still not worth it. So.
Well, for kicks and giggles, you know, what would be the bottom line for you?
I'd be happy to sit down and look.
Okay, that's why I thought that would be the answer, but we might have a discussion offline, because if we are going to go back to these markets and talk to them about this, we need to share that with them. Like, look, this is basically, we need to be talking here. And again, this may end up being where, all right, it's just not going to work out, but I mean, we got to be realistic about it. So yeah, so maybe we'll have a discussion later and throw out some numbers that we can share with them.
Back in the day, like, you know, seven years ago, we used to go out and do vertical line fishing and pull up 400-500 pounds in a day. When the numbers were out. Now, like Eric's saying, you know, you go out there and spend a whole day out there, you're lucky to get 200 pounds.
Okay. So we've done too good of a job with the tournament. We'll hear from the tournament people later. Thanks. All right. Chantil? Okay.
All right, so for those of us that have worked in restaurants, we know that a lot goes into food prep. And so my question is that if you're taking a lionfish that has hollow spines filled with venom, do we know the potency of the venom as it stands before they're despined? And 2, once it goes into a commercial frying vat, that's going to leach out into the oil. So then really the concern from the customer and the restaurant of the potency after it's been fried, if we could test the spine in some way, if there's even any fluid.
So you can use the venom, you can literally use those spines as a toothpick.
That's what I'm saying. It's like catfish.
And I think it's an education thing.
Yeah.
I think it's an education thing for the chefs on how can they best use, best price, and best prepare. I'm in marketing. That's what my full-time job is, what pays the bills. For the last 15 years, have been screaming, this is a marketing problem. It's not a problem with price. What is Chilean sea bass on their menu? $65, $70 a plate? What is swordfish? What are these high-end fish that the market has deemed, the restaurants have deemed, these are high-value fish? You know, I think one of the problems that we started out from a conservation side saying, we need to get these out of the restaurant. Let's make it inexpensive so more people will do it. I think that's bitten us in places we don't want to bite now, because now we can't get the price that we need. The restaurateurs are saying, I can only sell this for $20. Well, why? Who put it at $20? They did. And to an extent we did as well. So we need to back up and, you know, kind of determine that market price that the fishermen need to sell it to,
and then convince the restaurateurs that the fish has that value on a plate. So whether it's $12 a pound from us to get to $65 a pound to them, remember, we don't have unlimited supply as much as we have a problem. So if if a lower end seafood restaurant says, I can't justify putting a $60 fish on the plate, well, you're not our market. So let's look at the high end, the Jackson Steakhouse, the Dewey Destins, the ones that do have that level of fish on the market already. And then we educate them. We have them educate the servers. So when they're in front of that customer saying, hey, I've heard about lionfish, I'm kind of scared about it. That's an opportunity to explain how awesome this fish is to eat and how exclusive it is. And that's why that price. Anyway.
Go ahead.
We kind of, I'm going off topic of lionfish, but most y'all don't know I was an oyster farmer before, and we kind of had the same situation of where we were selling our oysters and nobody wanted to buy them for the 80, 90 cents a pop. Our newest tactic that I employed was going to the distributors and being like, hey, when you go to a bar, for instance, you have a lower wells and then you have the top shelf. So you can offer your red snapper, whatever you want as the lower tier. And then your top shelf would be the lionfish. And that kind of changed the thought around the oysters. And we were kind of hoping that would change the thought around the lionfish too.
I think it gets back to education.
Yeah. Well, two things I brought up, I want to hold out on this too. One of the things we're making the assumption, because we've been doing this since 2013, we assumed that everybody knew this, and that's what I need to hear is I'm assuming they know this, and I'm hearing now they don't know this. So yeah, we need to make those points. Yeah.
So those restaurants that you surveyed that were a little more concerned about trying to fry up a whole fish and serve it on a plate for whatever reason. My question is, do they serve whole red snapper on a plate? I mean, if you're comparing spines to spines, that's all they are. At that point, it's not, red snapper have large spines. They're just not 3 inches long.
The one of the restaurants that I surveyed, he did not want to actually serve whole lionfish. He thought that he had too fancy of a restaurant to be eating lionfish like they were brim. That's word for word what he told me. He said that you don't want somebody sitting there at their table picking off the stuff. Whereas the other restaurant I had surveyed, they were a little bit higher. And they did mention, yes, we do want to fry them whole. And he didn't really see, he was one of the only restaurants that I surveyed that really was like, okay, venomous spine's cool. Everything has got something wrong with it that we've got to go around and get around. Whereas I could see that changing. And no, you're right. Nobody really does serve a whole red snapper. I think that'd be pretty cool though.
Another thing I want to mention before I go on to the next question that popped in my brain when Brady said something about conservation. I remember talking, some of you might remember Dr. Chuck Adams with it's not Dolphin, Florida Sea Grant. He told me early on, he was very interested. He was our seafood guy. He's since retired. He was very interested in this whole thing as we were rolling this out, say 10 years ago. But he said, what's going to be interesting is that if this moves towards a popular product, your objective is to reduce lionfish in the Gulf of Mexico, and the last thing they're going to want is you to get rid of lionfish in the Gulf of Mexico. So you got to keep that in the back of your mind as we move this down the road. We got 3 minutes.
3 minutes.
Really?
Yes.
Wow. We never.
Look at us go.
Okay. Let me quickly. Okay, we got some questions in the queue. We may have to do that another time. Okay. Okay. One last thing, the big problem, and again, you guys can find us and give us more feedback before we go talk to these guys again. But the big problem with all of them was, again, the issue of the size of the animal and how much processing and cleaning it's going to take and the yield they get out of that. Has anybody who's having success selling these things and getting them in the restaurants have an answer to how to get around that problem? That's obviously number one.
Eliminate the fillet aspect of this. If
Okay.
they're concerned about the size of the fillet they're getting off the animal, if they're looking for something that's pre-cleaned, pre-packaged, no spines, no worries, unfortunately, yeah, we may not be able to serve up whole fillets of lionfish to people, but there's been restaurants in the past that have done dips. We could offer a plate. There are other ways around that you can still offer a product that is representative of what we're trying to do from a conservation standpoint without having to put additional restrictions on us and limit us in that fashion.
Yeah, like tacos.
Okay.
They don't have to be all.
Is that what's happening? Is anybody here working with a restaurant right now that's having success selling these things? I mean, is that what they're doing? Is all whole fish? Is that kind of how the process is going?
Okay.
Maybe share that with them. Yes.
Well, we're not working with a restaurant, but down in South Florida, there's a restaurant that the name of the restaurant is Lionfish Del Rey.
Yeah.
So, obviously that's their primary fish of choice. Now, we've gone there and not been able to get lionfish for the same reason that you're saying, right? But they have ceviche, they have lionfish bites as appetizers. So there's, the smaller fillets, you can do different stuff. It doesn't always have to be a whole meal.
Okay.
But I mean, their whole premise is lionfish, obviously. And, so there's a lot of workarounds. I don't work with them. We're with Zookeeper. So we work with some people that are commercial fishermen, and we might be able to get some of those answers for you.
We'd love it.
By talking to those guys, but there are workarounds to it.
Okay. All right, I guess in the sake of time, yes, Brady, go ahead. Yeah.
Along with the educational side of things, maybe what we do is we give them ideas on how to prepare smaller size lionfish.
Yeah.
When I was in the bar business, the distributors would come in with a new product, but they would also say, and here's how you can prepare it. Here's how you can make the most profit out of it. Maybe we need to take on that role of saying, here's how to do lionfish medallions with onions and peppers and a little bit of rice, something like that.
Yeah. Yeah, that's kind of what I've kind of made another pencil here. Just rethink how you guys want to present. Yeah. Yeah. All right. Yes.
So obviously it's coming up on Super Bowl weekend. The morning TV shows, they've been having some chefs come on and little things on how to prepare. And so when it gets time near some of the tournaments, prior to those tournaments, maybe get with the local news media and have a chef actually do one of those morning segments or multiple ones through the week with different things that you could do, and
I'd be down too. Yeah, I was gonna say you can do it.
I'd love to be it.
Yeah.
I'd love to do it.
right. So, anyway.
Thanks.
And there's our email addresses, Rick's and mine. Thank you all for everything. Thank you for all the responses.
Yeah.
I also want to thank Alex for all the great photos.
Put together something and go back to these folks and do a program for them.
Thank you all.
Thank you.
Awesome. Thanks, guys. Well, my name is Alex Fogg, and I will be your moderator for the rest of the day. So first up, we are going to have Emily Robichaux with the FWC Lionfish Control Team. She's going to give us some updates on Florida tournaments. And then after that, we're going to hear from Scott Ganello, and he is going to tell us about international tournaments. So Emily, take it away.
Yeah, make sure I don't trip.
Oh,
Trip anyway.
that's okay. We just got an
Got it?
arrow. Yeah.
Let's hide those controls
Nope.
up there at the top on the right hand corner.
If I've got a mouse, there we go. Whoa. Okay, I can see it.
And then it'll say the third one down, the very last one. Hide controls, but not-
Hide floating.
Yes, you're good.
All right.
Then you got to
We're
click-
live.
You have to click in the slide to advance.
Okay, we can move.
Yep.
All right. Well, hello, everybody. Thank you for making it out today, and thank you for the introduction, Jessica. Like she said, this is Florida invasive lionfish, kind of statewide updates. We're definitely going to get into the tournaments, but I've got a little bit of information about our program as a whole. So I'm with the Florida Fish and Wildlife Conservation Commission, specifically Division of Marine Fisheries Lionfish Control Team. So kind of start looking broad. These are our entire program initiatives. So while, yes, we really want to hit hard on that removal effort, we also want to touch on the education and getting people aware of the issue in the first place. We know this isn't novel at this point, but a lot of people still have misinformation or they're just not from around the coastline where they've ever heard of this issue. And so kind of our goal with that is to minimize these adverse impacts. We've talked about the descriptors of an invasive species, and a huge component of that is the conflict. And so we really want to minimize that. And so we do that with education through our outreach booth, which we will attend community events, festivals associated with tournaments, things of that nature. So if you're interested, we're welcome to have that booth at your event. We also do workshops in classroom invasion. Workshops are more of our adult focused, maybe in a dive shop or something else. And these are all based on entities requesting us to be there. Our classroom invasion is obviously school-aged kids, and that could look like running a kind of exercise with younger kids to dissections with older kids. And then we also have our Lionfish Educational Exhibit Program, which is a program that we've got set up to offer funding for creating an exhibit in a separate entity from the state. So just creating standardized education. We give you facts for you to use to put your exhibit together and then run creative with it. So we've got kind of an image down here on the far right of that exhibit, or one of our exhibits that have been put up over the years. And so then we're going to get into kind of what we're going to dive into today. And that's our control programs. And so that looks like supporting local entities, so events that happen around the state, and then our statewide Lionfish Challenge, and then other odds and ends that pop up around the state. And so specifically within control programs, our goals are removal efforts. And then kind of secondary to that, it is tracking recreational and commercial removals. And so this looks like local tournaments. And so both of these formats are achieving the same goal. We want to remove lionfish. These are aimed at it, but it's a little bit different approach. And so your local tournaments are pretty focused. Smaller location, smaller time span, that sort of thing. And then kind of in contrast, the Lionfish Challenge, which is our state-run FWC, pretty much statewide tournament, is 3 to 4 months long over the summer, and just hits a broader group of people over a broader time. And it's sort of catch-all that participate when you can, and if you can't, maybe next week. So to get into that, I also kind of want to talk a little bit about our Derby support. And so if anybody is interested in possibly hosting their own Derby, here's a little bit of information, but also to show you how we support the derbies that are already in existence. And so first of that is financially. We do have a program set up to offer funds for tournaments. We do have a minimum participation requirement of at least 10 participants for that derby. And so that goes into lionfish specific derbies, which are what we're more familiar with. And that's the targeted, but we also don't want to exclude other spearfishing tournaments that are open to having a lionfish category. So it may not be the whole goal, but if you're willing to add that component, we want to help support in that aspect as well. And then we're also willing to help offer technical support. So if you want to talk through some ideas, what works, what doesn't work, what's been seen somewhere else, we're happy to talk through that and give ideas and just play out what could be on the table. And then also attending tournament festivals. And so if you want to have that added component to your tournament, we're happy to be a presence there. We'll bring that, be the predator outreach booth, but also a couple other groups within our division or agency. We'll also have our artificial reef team sometimes, or other education outreach, because we know just because we're talking about lionfish, that doesn't mean that's our audience's only interest. So kind of broadening everything. And so if anyone is interested in this or wants more information, wants the application, anything like that, feel free to email lionfish@myfwc.com. So we're going to get into a little bit of the numbers and kind of what it's looked like over the last 10 years. This program was established within the agency in 2014. And so this is kind of that tracking component. It's secondary, but it's interesting to look back over the last decade and see what it's looked like. And so this is our total number of tournaments. I hope y'all can read it in the back. If not, we've got total number of tournaments going up to about 30. And then every column is 2014 up to last year, 2013. And the subsequent graphs will also be that same display. So what we look at here is kind of also tracking a little bit of that peak that we've seen with the invasion curve. And then a little bit of tapering off. We took a really, really hard hit in 2020, and then have slowly been bouncing back from that, but definitely are not at the numbers we were beforehand. And that can have a lot of things that play into why that is. So that's just kind of keep those numbers in your mind when we go forward to number of participants on the left in green and number of lionfish harvested on the right. And so we kind of see that our total number of participants tracks with the number of tournaments. So if we're offering them, people will show up, but if we're not offering them, we're just not really seeing that huge boom with fewer tournaments. But interestingly enough, our lionfish harvested don't necessarily track with that. So while we're having fewer tournaments and fewer people per se, we've also had an increase in growth. So the people who are participating are very passionate about it. And so that's kind of something that we want to capitalize on. If they're really interested, let's be pushing them to tell other people about it and get other people involved, because we want that participation higher. Because if we can get these numbers with fewer people, we can get even higher numbers with even more people. And so kind of then the total number, we're just shy of 200,000 fish that have been tracked over the last 10 years through these statewide tournaments. I will also note the Lionfish Challenge is not a part of any of these numbers. These are all independent local tournaments in these numbers. So then we'll kind of go on to the contrast with the Lionfish Challenge. Its focal point is always during peak dive season. So it's going to be 3 to 4 months over the summer, kind of targeting that effort. And we have partnerships with dive shops. So kind of something that I didn't get into is a really great aspect about local tournaments is that face-to-face value. You have people working the tournament or the festival, so anybody interacting with the tournament has a direct way to talk about what's going on, to get updates, to be put in contact with other people. And that's something we really don't have with the Lionfish Challenge. Because it is statewide, we have checkpoints around the state for our recreational participants. So they do not see us as an agency. They see the dive shops that then work with us as the agency. So they're kind of our middleman. They are our boots on our ground, the people that our participants are seeing. And so that's a great partnership with them, and we can relay information through them. And that's also a great way to have community involvement and increase business. I have had people around the state saying, well, I really want to be involved with the Lionfish Challenge, and I typically go to this dive shop, but I got to turn in tails over here, so I'm going to go get my tank fills over here. So it can draw interest in other directions. But we have product-based incentives, which are physical prizes that we will hand out to people as they earn certain tiers. And then we also have raffles. So we'll set number of fish to turn in. And once you reach that number of fish, you'll get entered into that raffle. So sort of an extra layer and bonus on your tiered prizes. And then we also have resource-based incentives. And so since we are a state agency and have that broad aspect, we've been able to write executive orders each year to allow divers to obtain an extra spiny lobster each day during sports season. And so this is last year's coin, which is just, again, kind of different aspect that we can factor in being at the state level. But I will also make a call for, we're always open to sponsors. Any new checkpoints, any new products people want to air, things like that. Happy to talk, happy to work on that and have those products as a part of our competition. So we'll get into some of the numbers with the Lionfish Challenge. And again, this is specifically the Lionfish Challenge. This does not have local tournaments as a part of it. And so I'll just kind of let you look at the graphs for a second. I will say the green is our recreational division and blue is commercial. And so kind of following up this whole conversation about marketing and marketability and commercial effort, this is really what we've seen within this competition. And we know that there are people that do have commercial efforts that don't necessarily take part in this. But it's really interesting to see over the last decade kind of how some things have shifted. And so we've seen a contrast and a change in recreational and commercial effort harvest by year. And so where we used to have predominant commercial effort, it's now changing to predominant recreational effort. And we had our low this year of, I believe, 11 commercial participants, where we had a record of 275 recreational participants this year. And so just looking at the graph on the left, we pretty much had steady increase with recreational and steady decline. So that's something we really want to focus on this year, is potential commercial interest and making that viable tiers and interest and worth their time. And so then we're also seeing that on the right side with participants. They track the same of so many more people and kind of a shift with less. So that gets into kind of what's next. What does this look like for us? So local derbies, if you look at them around the state, are derbies that are maintaining consistency are very much Panhandle and then South Florida around the Keys. And so those are entities that are very committed. And so we're familiar with those around here. We've also had a handful of new tournaments that are popping up in interest. So we really want to work on supporting those and giving them the tools that they need to continue past year 2, 3, that kind of thing. So we're working on finding ways to support them and what will be best for their area, what we want to see. And then we also want to see derbies in new locations. So we're working on reaching out to other entities in areas that we're not seeing tournaments happen, or maybe we've seen them in the past and they've dropped off. So expanding what is available and then increasing the participation. And then at the state level with the Lionfish Challenge, we are looking at trying to refresh prizes. It's hard when you have a niche audience and you're looking at limited with, you can't do anything with sizes and things like that. But we want to make stuff that draws people in and interests them. So if anybody ever has any ideas or wants to talk about that stuff or wants to see something new, I'm all ears. And then this year, a really big focus point is modifying those commercial tiers to really make it feel more attainable and more interesting and have a draw, because we know, like we're hearing, we don't feel fully compensated by the dollar amount that we're getting for that commercial market. So hopefully this will kind of bolster that. And then kind of wrapping back into our entire mission goal of the program is maintaining consistent and accurate education. And so that's for some of our events that we're attending and things like that. But we really want people to not only know about the issue, but have the accurate information and what next. How can we handle this? How can we get involved? What does that look like? And individual interest levels, things like that. So if anybody has any questions, interests, wants to reach out and contact, I've got program information in my personal. Lionfish is just a little bit easier than spelling my last name. So easiest way to reach out is by email, lionfish@myfwc.com. But I'm happy to hear from anyone, everyone. And if you have anything that you'd like to see, I'm all ears. So yeah. I don't think we quite have time for questions, but we can pass on. And if we have time after them, come back.
15 minutes. So possibly during break 2, I would suggest asking questions then.
That's fine. I'm around. We're good.
For Scott.
Thanks so much, Emily. So next we are going to have Scott Ganello. He is with Lionfish Central. He is virtual, so we will hear from him through the computer screen. I don't know what to do from here.
Can you hear me? Can you hear me?
There we go. Okay. Okay.
Alex, your voice is a little higher pitched, but can you hear me?
Okay, well, you do this and I'll.
Can everybody hear me? Hello, can you hear me? Anyone, can you hear me? Hello, can you hear me?
All right, Scott, can you hear us okay?
I can hear you. Can you hear me?
Not yet. We hear you barely. I've got to turn your volume up. And what we see right now are your notes and what you're sharing.
Okay.
So if you could switch displays to your actual presentation.
Can you see the presentation now?
Yes, I can.
All right. I will try to speak louder so you can all hear me.
Okay. Is
Volume
that
okay?
Yeah.
All right. Well, thank you everyone for having me. Emily, you're a hard act to follow, but I'll try my best. My name is Scott Ganello, and I'm the founder of Lionfish Central. We started the nonprofit to kind of fill in the blanks for the lionfish community. And we got to work right away developing an app that is being used in about 30 countries. It's free for all divers and anybody interested in lionfish. It's called Lionfish Patrol. It tracks depths, bottom time, how many fish they caught, what time of day, what type of poundage, weight, kilograms. And we do a lot of converting of stuff. So if you tell us how many fish you caught, we'll break it down to kilograms and pounds, sizes, metrics, standards. So, and there's a leaderboard. And that's become very useful for some of our tournaments overseas, where we can look up specific divers from specific countries and help coordinate tournaments for their area. So there's a lot that the app does, and it's been very useful. And we're in about 30 countries. We also created a video game for kids because we want to teach children that for every lionfish you remove, you add more fish to the game and to your reef. We did a comic book. Some of our travels, we did do a lot of podcasts since we're on all these little islands talking with all the lionfish hunters in the area. We do lionfish expeditions, and we have this two-person submersible that's coming back by March 1st that will get back into the water. But that's the boring part of the project. So I want to talk about two different tournaments that we've done throughout the world. But just to kind of give you a background, we either run the tournaments fully, or we'll be part of a team. Like Antigua, I was with Fogg and Dr. Giddings and Lionfish University and Megan and Phil, just a whole group of us that kind of went in and with a little bit more better money from Antigua and that they raised. It was a great big festival, and it got the locals involved, and it was just a great tournament that was put on. And so other ones we've done is Aruba, Belize, Curacao, Panama, the Panhandle with Emerald Coast. Helped South Carolina Aquarium, kind of played some behind the scenes, kind of guided him when he first had his first one, got him fillets, fish for his cooks. So we either can be totally silent behind the scenes, or we can take the ball and run with it. So one of the ones we took and ran with was Curacao. We didn't get a lot of data from Curacao, and we wanted to create a tournament where the divers had to log their fish on the app. And then there's leaderboards to see who's winning. And we didn't want to just do a small one. We wanted to do a 3-month one, because after 3 months, they've been kind of conditioned to use the app regularly and keep track of it. And then they've kind of, built up their total on their account. So, and it puts them on a leaderboard for their country. And Lizette, we all know Lizette. And if you don't, you need to know Lizette. She runs a business called Lionfish Caribbean, and she has 2 tournaments a year. And I worked with Lizette to create our last day of the tournament on her 1-day tournament. So we had a double festival at the end, twice as many prizes, and everybody that caught fish on her tournament that were in my tournament could use those as well. So there was more of an incentive for everybody to be using both my app tournament and the Lionfish Caribbean. She makes jewelry out of the fins, and she needs more fins all the time. She has a restaurant, and now she's serving lionfish, I think, 6 days a week out of her restaurant. And she has a gift shop. So she buys the fish from all the divers. And she single-handedly has done just an amazing job for Curacao in the sense that they patrol the reefs regularly, and the divers make money from it. And she willingly and gladly pays them and sells a ton of jewelry around the world. So working with her was a easy way to get the tournament up and running. She helped promote it, and she helped tie it in with hers. And over 3 months, it was a pretty good success. We had about 3,000 lionfish caught in the app that they logged. We had a ton of app data, which is great. She bought all the fins. She got all the food. We brought in cash prizes. She had her cash prizes. And Zookeeper, a great supporter of what we do, is always giving us, zookeepers and product to bring as part of the raffles or the prizes, the giveaways. Lionator Pole Spear, he always donates some stuff. And it's just it was an easy way to take a tournament and build upon what's already kind of on the ground and running. And some of the bigger islands that have it down pretty well, it's pretty easy to do that. On a personal note, the woman on the left in the middle, she's about 15 years old, Fada, she's holding the pole spear. I made sure she won that because she's a free diver, and her spear broke. And then about a year later, 6 months later, she was on a NOVA program about lionfish. Fogg was on there, Jessica was on there, Lizette, but Fada was the free diver. She free dives after school, 15 years old. She free dives after school and sells the fish to Lizette. And there she was free diving on this NOVA special with her new pole spear that she won at the tournament. So it's nice to see it come full circle. It's a great community. So Curacao is one of those islands that is just doing really well with lionfish, and Lizette is the driving force behind that. Now, the other one that we've worked with that's been kind of a success is Tobacco Key. And if you've ever been to Tobacco Key, you'll know that this picture doesn't do it justice. It's a small little island about 15 miles off the coast, right on the Barrier Reef. Population's about 30, 35 people. It's a very small island, and everything has to be bussed in. You want ice, it's got to be not bussed, but boated in. So you want ice, it's got to be boated in. Water, food. It's pretty much off the grid, although they do have generators on the island, some solar, and they catch the rainwater. But it's not the easy way of life, but it's a beautiful, beautiful island. And the gentleman that was running the marine station is James Troughton. He's a marine scientist, and he runs the Tobacco Key Marine Station for the last 5 years. His job is to bring students in, interns, and teach them all about marine science. He's developed tons of courses for them. And last year alone, they brought in over 1,200 people through their doors by the end of September. But one of the big programs that I helped him set up was the lionfish hunting and the lionfish tournament. So he reached out to us, and we basically kind of helped him put it all together, gave him all the tools that he needed. We brought in a whole bunch of stuff, prizes and stuff that they needed on the island because it's so expensive to buy and ship there. So we, had a couple extra suitcases full of stuff that we brought in. We donated cash and prizes. And again, Zookeeper steps up all the time, and Lionator steps up, and Stingmaster. And the first year we did that, we did it on an hourly basis. So you'd go out for a couple hours, come back, drop your fish off, go back out, come back. And they only caught 215 fish, but that was with just a handful of divers, and most of that was free diving, which was actually a really big success because all the students took the time to, dissect them, weigh them, do the mouth measurements, do stomach contents, and record everything. So the interns really do some great work with that. And out of all the tournaments I've seen and been to, they do more for gathering data, I think, than anybody, right down to the stomach contents. And they documented over 50 different species that were being, decimated on their reefs over the past 3 years. So they do a great job. And it's the first one we kind of spearheaded. And after that, they've been doing it on their own. And the last one was this past September, we went and helped out as well. And it's a really incredible program that they put on. And you got to remember, these little barrier islands out there are all dotted out, throughout the coast of Belize. This one brought in a lot of divers from the mainland and from islands very far away. And this was a huge festival. They caught just under 700 fish, lionfish. The interns got through all of them in a day, documented everything. And what happens is the locals, they only make about $2.50 an hour for minimum wage in Belize. So when we come in with a lot of money and a lot of prizes and a lot of handouts, a lot of cash prizes, they make more in one day doing the tournament than they might in a month or two on their current job. So this one was a very large one, and we had people coming from all over to join in on this one. And, so the big cooking contest at the end, there was probably 200 people on this island that holds about 35 people on a regular basis. So it was a huge event. And, I talk about community here, right? The community bringing people to this island and getting together with friends and family. One of the interns was from Hopkins, and her father was there, her cousins were there. And to see all these people get together and have this sense of community and do a great job keeping their reefs clean in that small little area, it's really inspiring to watch and to see the prizes going out. They had, alcohol vendors there with their Belize rum giving prizes out. There was jewelry making going on over there. There was the fins were being given to a jewelry maker on Key Cocker, hours away. It was just a really great thing. And to see the community all come together like this was really inspiring. And, over the years, I've worked with a bunch of people from Belize, Celso, who is with the Lionfish Brigade. He showed up and his team showed up, and they're the only ones that have a permit to hunt lionfish on the barrier reef side. So it was really great working with him. And even Polly Wood, I missed the chance to meet her a few years ago. She was in the same crowd, but I didn't know who she was or didn't know what she looked like. And as a joke, I texted her and said, "Hey, if you're in the area, stop by." And she wrote back and said, "Yeah, I'll be there shortly." And, here she comes. She pulls up and I got to meet her. And it's such a great community in Belize, and they do so much to protect their reefs. So it's really, your money can go a really long way putting together a great program for the locals and help get the data. So we get a lot of data from there, help keep the reefs clean and try to keep them lionfish free as possible, but help the community. Just giving them new zookeepers and new spears helps those divers, because I've seen their spears when I first went over there, and they were homemade, and it was scary what they do to get there. So these little tournaments go a long way to help protect the reefs along the Belizean coast. But what also is nice about this is gathering lionfish to be able to be exported as a product. There's a restaurant down in the Florida Keys. I know you were talking about restaurants in Florida. There's only one restaurant that I know of in probably the United States that has lionfish on the menu every single day of the week, of the year, all the time. And that's Castaway in the Florida Keys. And is getting harder to find lionfish because of all the great work FWC is doing, and Reef and all these other tournaments. But I think a couple months ago, he just brought in 600-700 pounds of fillets out of Belize. So again, we're always trying to work with these different countries to create ways for them to make money catching lionfish to support their family and to keep the reefs clean. So it's a really good way to work with these smaller countries. Now, we've done some in other islands that it just didn't go. It didn't work for political reasons. Well, this group doesn't want to work with that person, or, some of these islands can be really small and it's kind of hard to work around. And others just, kind of drop the ball on it. So you kind of have to pick and choose. You throw them out there and hopefully they go. You give them as much support as possible, but it's not guaranteed all the time. But Tobacco Key Marine Station, and by the way, James just left there after 5 years, and we just hired him on as our new director for the new project that we're starting, with his teaching abilities and his efforts that he's done around the world. And that is our Marine Science Odyssey. Our Lionfish nonprofit is not going away. We're just kind of including it into this new learning platform for everything marine science. We're currently working with NOAA, with the Coral Reef restoration program, working with Mission Iconic Reef to put their information in there. It's a free platform for anyone that wants to learn about marine science. And if you have a nonprofit for Lionfish, Brady, or FWC, if you want to get your information out there to help have more people learn about what you all do. And it's not just lionfish, it's everything, coral restoration, turtles, nudibranchs, whatever you want. But that's where we're headed. And it's not being marketed yet, but it is live. So go take a look, sign up. It's free to join, and coordinate with us and get your information out there as well. And with that, my time is almost expired. So if there's any questions, or if you want to continue on to your break, because I know it's lunchtime, have at it.
We've already been to lunch. We went to lunch a little bit early, so we have time for probably 1 or 2 questions real quick. Does anybody have a question for the other Scott? Sir, we appreciate it. We don't see any questions here. If we do, we'll certainly get a contact with you. I was able to share that contact information for your website and other contact online. So appreciate your time and your help today.
Great. Thanks for having me.
You're welcome.
Take care.
Just said 5 minutes ago. All right, so we are going to get back into the tournament updates. First up, we have Tim Robinson with Zookeeper. So please come on up here and tell us all about your wonderful business and what you do. Oh, I think just hit the arrow. All right.
All right. Well, first off, I want to thank everybody for the opportunity to talk to you today and give you some updates about what we're doing at Zookeeper. You've heard a little bit already from some of the other presentations, but I'm going to start off with telling you a little bit about where we came from, where we started. This will sound a little bit like an infomercial for a few minutes, which it is. And then we'll talk about what we're doing as a company to help promote the all the events and stuff that's going on out there. So where did it all begin? So with Zookeeper. So it began in Curacao. You heard Scott talking about Curacao. Well, that's where Zookeeper actually came from, right? They needed a practical solution to the problem. What are they going to do with these venomous fish so that the divers that were retrieving them didn't get, stung? So Ali El-Haj, who is the founder and the inventor of the Zookeeper, this is kind of one of the first off prototypes, gray PVC pipe, right, with a bunch of different components to it. And he had some friends that worked with him down in Curacao, and then Ali moved here to the States, and he ended up with a product that you've seen on some of the videos or some of the photos from the presentations today. It was a white PVC tube. And then Ali wanted to take it to the next stage, which was to introduce the clear pipe, right? So everybody could see what was in the Zookeeper. And then next he wanted to have different sizes, right? So some of the folks in this room, they know in the beginning, there's the commercial Zookeeper, it was like carrying a 55-gallon drum down underwater with you, fill it up with as many lionfish as you could get in it, and then figure out how you're going to get it up to the surface. But these were more designed for the recreational divers, right? So we took over the company in 2021. I bought the company from Ali, and basically one of the things I wanted to do was make what I felt were some improvements that were needed, mostly in the sense of diver safety, right? So I come from a background where safety is important. So there was a couple things. Number one was the Zookeepers had a lanyard on them, but they didn't really have a handle, a way to hold it. So we just simply added a handle, which was an option that Ali offered. We just made it on every Zookeeper. We added it to every Zookeeper so that the divers, had something to hold on to. I dove with plenty of people, and I would see them put their hand on the rim of the Zookeeper and then push the lionfish right past their hand, right? So it was just a matter of time before they were going to get stung. So we added the handle. The second thing was, and this was really two reasons, we wanted to change the funnel, right? And if you have, and I know there's still some zookeepers out there with the old funnels in them, but they were basically a car funnel that had to be modified. And first off, just to modify that funnel was something I wanted no part of, because we build these zookeepers. We have a facility in South Florida where we make them, and slitting and cutting down the funnels and all that stuff was nothing that I wanted to have a part of. So what we did is we designed a molded funnel. And if you have any experience with molds, plastic molds, they're very expensive. We'll probably never retrieve the cost of this over time, but it was, again, more of an interest of making the product better. So a lot of guys know that you take that fish, you shove it in the zookeeper, and you're pushing the funnel right down in there too, right? Because there was no lip on it or anything like that. So we designed the funnel with a lip so that it would sit on the edge of the zookeeper, and you really can't push it into the zookeeper. They're a little shorter, gives you a little more room, so on and so forth. So those were the two real big design changes that we made, or not really design. Adding a handle wouldn't be a design, but two of the changes we made that we really felt added a little bit to the zookeeper. Then next came the most important thing, right? Which is, while you're doing it, you got to look good, right? Everybody knows that. So that we had to do something. So we started adding colors, right? And we came out with the black zookeeper and with the two different color wraps. Now, I know Ali had a wrap early on, but that was just because he was trying to cover up the white PVC pipe. These wraps are still super popular today. People buy them and order them all the time. But we introduced the black, and people went crazy. People that had white zookeepers were buying black zookeepers. I'm like, oh, we could start selling a lot more zookeepers. We just keep adding colors, right? So the girls were like, hey, we want pink. So we had to, take care of them. We added the pink, and then we came out with the teal. And these are the four colors that we now carry today, right? And then we're like, okay, now we've started a trend here. We got to keep it going, right? So we didn't stop there. Now we came out with the limited quantity yellow, right? Which was a big hit at the DEMA this year. We introduced it at DEMA and came out with the yellow. And of course, now we're getting all kinds of suggestions of what should be the next color. Alex, you want to scream it out? All right. So that'll be a secret, right? We'll let you know what that next color is going to be at DEMA next year. So the yellow is a limited quantity. We ordered enough to make about 400 zookeepers in yellow. Once it's gone, and we'll come out with another color. But as you heard from some of the other presentations, Zookeeper is very involved with other organizations in supporting their events, right? Whether it's the big Emerald Coast Open or the local derby that's being done on a Saturday morning. And we were giving away so many zookeepers because everybody contacts us that wants a zookeeper. We decided that we were going to introduce another color that was going to be our giveaway color. So you can't buy this zookeeper online. You can only get it at an event, and it's our blue zookeeper. Okay. So this is some that we actually personalized for a derby that we did, or we participated in and sponsored down in Belize, not with Scott, although we support Scott, and you could tell from his presentation. We were in San Pedro in Belize. And so these are the blue zookeepers. And now what we're doing is when we give away zookeepers for an event, they're going to be the blue zookeeper. And then when we see blue zookeepers out there, we'll know those folks got them from an event, right? So it's kind of cool. We can customize them. We customize the wraps. We do a lot of different things. So that's kind of where we're at with zookeepers. That's the end of the zookeepers. But then we started, Ali had a vision when I bought the company. He actually had the components, but he wasn't I know he made a few prototypes, but he wasn't making sling spears. So we went ahead and we finished and we added the sling spear. And what we were doing is we were listening to what the divers were telling us, and we're divers as well, was, you know, there's so many sling spears out there, but nobody's got one with really got a grip on it. So we have our sling spear that we have a grip on, and they're super popular. We really can't keep them in stock. We hand make them, and they're very popular. We have them in white, black, red, and maybe some other colors to come. But, and then we have our spear tips, right? These are the Zookeeper spear tips, which is our traditional paralyzer with no barbs. We have our quadrilizer tip with the extra long tip in the center. And then we just recently introduced the paralyzer with barbs. Again, listening to the diving community telling us what they wanted, right? They wanted a tip with barbs on it. So we introduced that and have that now available. Of course, we have a full line of apparel, shirts, shorts, hats, gloves, anything that you want to wear to show your support of Zookeeper. But the biggest thing is, and I'm going to move on, get off the infomercial thing, is what we do to promote the lionfish removal, right? So right after I bought Zookeeper, I just kept hearing it really started with, hey, everybody's like, hey, you know Alex Fogg, hey, you know Alex Fogg. And I'm like, no, I don't know Alex Fogg. And so finally we met at DEMA in Las Vegas in 2021. And 2021, I guess it was, yeah. And I met Alex and you know, we hit it off and we started talking about this event. And the next year we were a sponsor of the Emerald Coast Open. And, you know, we started out, I think, as a silver sponsor. Last year we moved up to a gold. And again, this year we're sponsored as a gold.
Wow.
No, we're going to stay gold right now. Then we also work with Reef down in the Florida Keys. We sponsor their events. So we're a big sponsor of that. And then last year we worked with FWC and sponsored the summer challenge that Emily talked about when she was up here, and, you know, loved being a part of that as well. So these are the three big kind of events that we did. But then now we move on and we have the South Carolina Seaquarium. They have a big lionfish derby that they do. They call the entire month of July, they call it Lionfish. And so we became a sponsor of that event, which turned into us actually sponsoring their exhibit at the aquarium. So these are some of the plaques that are on the exhibit at this aquarium. So we promote their event, the Lionfish event. We support obviously the exhibit itself, and then their research and stuff. We provide them with zookeepers and sling spears and all of the stuff for the research and stuff that they do as well. And then this is just a list of some of the events. There's the Emerald Coast Open, the Reef event, the Pensacola Lionfish Challenge shootout that we sponsored last year, the FWC. And then it kind of trickles down to some of the smaller local events. We've done events in Miami, Deerfield Beach, San Pedro, the one I was just talking about. We did an event with Toucan Diving down in Bonaire, Ocean Encounters in Curacao, Finn's Dive Center in Key West, North Carolina, and then again, Utila. We sponsored an event down in Utila. So you can see we're all over the place. We get contacted every week for people that are doing events, you know, can we sponsor? And sometimes it's money, sometimes it's just product, and sometimes it's a combination of both. But again, we're out there trying to educate the public and, you know, get people involved. We do DEMA each year. Then we have our indoor display, we have a outdoors display we do that we can take to all the events. Of course, at the Emerald Coast Open, we have a tent with product and stuff. And it's, you know, again, just an effort for us to support these events and obviously continue to grow the brand, the Zookeeper brand. We are, it's a family-owned business. We're a group of divers that are instructors as well. So we teach the PADI, we certify folks with the PADI Lionfish specialty course, which if you dive in certain parts of the Caribbean, Bonaire being one of them, in order to go hunt lionfish, you actually have to have the certification. So we do that stuff. And, we also have been for the last couple of years promoting the use of the Zookeeper for pier cleanups and stuff. So if you've ever been involved in any of those events where you're putting fishing line, hooks, knives into mesh bags, it's a nightmare to get that stuff out there. So now if you just put all that stuff into the Zookeeper, you unscrew the cap, you dump it out, you're done, you're gone, right? You're on your way. So we're working in different areas to, educate folks about the lionfish, promote people getting involved in it. And then also on the cleanup portion, taking care of the reefs and the ocean. So we welcome you to follow us on all this social media. We are out there every day posting content. We have a lot of people from all around the Caribbean and stuff that tag us, and likewise we do the same back to them. And then lastly, we as a group, we do trips. So for example, in Curacao last year, we went down in April and we took about 17 divers with us. And then on that trip, we shot 611 lionfish. So we're continuing to do these trips. We're going down to Curacao again this April, and we've already got 22 people signed up. So again, we're teaching, educating, and we have people from all around the United States that are joining us on these trips. We got people coming from Texas, Washington State, up in the Northeast. So, we're just trying to get the word out there and make people aware of what's going on and what we need to do. All right. Thank you, guys. And I've probably gone over my time. If you have any questions, we can get to them at the next break.
Thank you, Tim. That was wonderful. For those of you who participate in our pier cleanups, Zookeeper has been generous enough to help sponsor those, and we will be using Zookeepers instead of those horrible mesh bags. So that is very exciting for those of you that participate. You know the struggle of pulling hooks out of a mesh bag. So next we are going to have Andy Ross come up and talk. He is going to talk about the Emerald Coast Open. That is the world's largest lionfish tournament. And it's actually hosted right here in Destin, Fort Walton Beach. Take it away.
Thank you. Well, it's hard to follow up a pink Zookeeper, but I'm going to give it a try there, Tim. Let's see. This is not an apple, so spacebar. Yeah. There you go. Anyway, welcome everyone. I'm Captain Andy Ross. I'm with Niue Dive Charters. Run a scuba diving charter out of Pensacola, Florida. We specialize in spearfishing and lionfish hunting. We also do a lot of recreational scuba diving. We take people tech diving to the Riscony. I'm also the tournament director for the Emerald Coast Open. That's a big word, a big title right there, but that means I'm a jack of all trades. I get to do a little bit of everything for the tournament, get my hands in a little bit of all that. But I'm also part of the Gulf Coast Lionfish Tournaments, which is really the back, it kind of runs behind the scenes for the Emerald Coast Open as a management crew right there. We kind of behind the lines. We track the sponsors, we track the registrations. We take fish and move them from point A to point B for the restaurant week. We handle getting prizes in, we handle writing checks, doing 1099s, all that stuff that goes along with running a tournament that you don't think of. That's what we do to make sure these things run pretty smooth. Okay, so just the the other day, I kind of realized that I've been working with tournaments for 10 years. I did our first tournament back in 2014, was actually at Florida, at the Floribama on the Florida and Alabama line. We did 4 events out there. And that's where I first met Alex Fogg. He was still going to school where he was learning about lionfish. We're all learning about how to do prizes and how to give away prizes and all that. From there, we moved to downtown Pensacola, and we did a tournament there, and we partnered up with FWC and did the Lionfish Removal and Awareness Day. And that kind of spawned several years of tournaments there. And eventually in 2019, we migrated to here to Destin, which is really a pretty good venue. And along the way, we've met a lot of great people and had a lot of great times. And it's been a pretty phenomenal ride. So coming up this year will be our 11th year of doing tournaments. I've partnered with Alex and some other ones. But just to show you some of the numbers here, in 2014, we got like 5,200 lionfish. 2015 and 16, the numbers are a little bit off because I don't think we kept really good track like we should have. We lost a couple of those. We did, I think 1,500 fish is probably light. We probably did more like 4,000 or 5,000. 2016, we did 9,000 fish. 2017, another 6,000 fish. 2018, 13,000 fish. Oh, we're starting to pick it up a little bit there, right? Then we moved to Destin, and our first event here in Destin, we had 189 participants and got almost 20,000 fish with 19,000 fish. 2020 was a little bit off because we had a COVID year, and I think we didn't really do a tournament that year. The only reason that there's fish numbers up here is because we had enough people shooting in the pre-tournament, and we decided to give away some prizes based on what individuals turned in, but we really couldn't have a regular gathering because it just wasn't really the thing to do. It was a very tough time that year. I think everybody remembered that, but we did manage to get 4,600 fish in, and 41 people were out there shooting fish kind of on their own, away from everybody. So we weren't in contact with anybody for COVID. 2021, we got back on the wagon again there and got 10,000 fish. 2022, almost 14,000 fish. And then last year we did almost 25,000 fish. Those numbers include what we did in our pre-tournament, which runs from February 1st till right up against the 2-day event. So that's really what we consider part of like a 2-part tournament. But man, that's a lot of fish. That's over 108,000 fish in the last 10 years. So my hat's off to everybody that participated in those 10 years. It's been really a phenomenal time, and we're real proud of that. We're real proud of that. So you can see the numbers are a success, very fantastic. But these numbers don't even include what Emily was saying. I think the Lionfish Challenge in itself, without the other tournaments, was like 190,000 fish, and I think the slide she showed us that overall tournaments in the last 10 years did about 200,000. So we got probably, at least in the tournaments I've been doing the last 10 years, we've gotten probably half of the tournament fish. So between that and the Lionfish Challenge, that's about 400,000 lionfish. And there's a lot of people in this room that have been shooting in those last years, and we've done a great job. So been fantastic that we could get a lot done just within tournaments. So that's it's been a good thing. So this will be our 11th year of doing tournaments. One of our greatest accomplishments is the Emerald Coast Open. It's the largest tournament in the world. 2023 was the largest prize giveaway ever. We gave away $65,000 in cash. And I remember because I wrote a bunch of checks and did a bunch of 1099s and junk like that. We gave away, made a lot of people happy. Of course, they worked really hard at that to get a lot of fish in. We gave $30,000 in table prizes, which included everything from pole spears to zookeepers to wetsuits from the dive shops. We got tons of prizes on that table. And we also started a couple of years ago, we've got a monthly drawing in. So if you were to turn lionfish in during the pre-tournament, if you even turned in one fish during that, let's say the month of February, you were eligible to get a monthly prize. And we gave away like 5 prizes a month. Usually in February, we'll give a jacket and a wetsuit and that kind of thing. But we gave away prizes every month. So we probably gave away another $4,000 or $5,000 in prizes during that period. Okay, some of the things that were we conclude that were contributing factors for our success was the over 60 sponsors that we had, firm sponsors that were able to donate prizes and prize money and be there to help us out at the count station, because that in itself is a chore, and all those volunteers for sponsorship. We had 144 participants last year. And in fact, the last 3 years, we've only gotten to 144 or 145. We just can't seem to break the 145 number. Although we did a few years ago hit 189 participants, which is quite a few. That's a lot of divers. That's a lot of support there. So the participation is very important. The venue was very important. We were at the Harbor Walk here in Destin. We had our tournament captain's meeting and prize giveaway right there at AJ's. That was fantastic. And we can't thank our volunteers enough. We had Florida Sea Grant and FWC and Reef and several more that helped us do some volunteers. And then also I couldn't do it without our staff. Okay. I consider my staff, Alex Fogg, he's kind of our ringleader. He's been pushing us along. I've got Brady Hale. He helps with the tournament. Scott Ganella, which you saw him give a presentation here recently. Jessica Velick and Mike Norberg and several other accounting people and people that added up a lot of the numbers and all that. But I want to really thank our staff too for helping out, and all the other people that really put this on, because to do this kind of an event is really, it's definitely a team effort, and there's no way we could do it with just one or two people. We definitely have to have a group of people here. It's become a very much of a family and friends event. We got people from all over, actually all over the world come here. A lot of the local people that come here are from like Florida and Alabama and Mississippi and Louisiana, even Georgia. Nationwide, we get people from Texas and California and New Jersey. I just had some people call me from Pennsylvania that wanted to bring a team down, and somebody else from Tennessee that wanted to bring a team down for this upcoming Emerald Coast Open. So we got more people coming in. And as well, we've had international travelers come down. We have people from Belize, Carousel. There's some people from Carousel last year. We're trying to just basically waiting for the Jamaican bobsled team to come in to help us out with it. We got everybody coming in. It's a great event. So we get a ton of people coming in. Anyway, the trend to date, last 3 years, we've been right around 145 participants, and Alex and I are both pushing to get past that 145. I don't know what it is about that one number, huh, Alex? We want 150, but we haven't been able to get past that. Okay. We've been averaging 60 plus firm sponsors every year for the past 3 years. The prize pool is getting larger. The top teams seem to be rotating every couple of years. We've got some great teams, but as one team either has won all the money they wanted to win, they decide another team comes in, and so we've got a lot of great things going on with those teams, and we've had a lot of international draw to it as well. Right now we've got 38 registered participants with at least 20 sponsors on the books for helping us out. The pre-tournament kicked off on February 1st. We've already had over probably close to our record, 425 fish have come in for the pre-tournament. And we're looking forward to another fantastic event. We're always looking for great volunteers and sponsors. So just keep us in mind. And we are very proud parents of this Emerald Coast Open, is our kid. Thank you. Questions? No time? Huh? 1 minute?
Yeah.
Anybody got any questions?
When is the 2024 Emerald Coast Open?
Oh, it's sometime the weekend after Mother's Day.
And
Anywhere there's lionfish, you can shoot lionfish and you can bring them to us. If you decide to money, I guess we can't stop it, but that gets kind of pricey, bringing lionfish in. Any place that there's lionfish are available, you can shoot them over. He's got an idea. Anything else?
Oh, Vice President. Second.
Okay.
Thank you.
Make sure I get on.
All right. Next up, we have Brady Hale, and he's going to talk about the Pensacola Lionfish Shootout. And then after that, we're going to get into the education and outreach portion. And Brady will be talking again. So I'm not going to step up in here between there. You can just go right
jump
into
right
the
in.
next one.
Great.
That one, you just hit the arrow button.
All right. Well, thank you very much. My name is Brady Hale. I'm with Gulf Coast Lionfish Tournaments. Captain Andy and I run a lot of tournaments around the state, and these are two of our largest ones for sure. So the Pensacola Lionfish Shootout last year, October 20th to the 22nd on Pensacola Beach. I want to contrast that from Emerald Coast Open. Pushed the wrong button. Do it this way. Okay, so this was our first year back in Pensacola. Like Cat Manny talked about, we were in Pensacola for a long time. We've moved over to Destin, and as we say, absence made the heart grow fonder. So we heard a lot of talk in Pensacola about we want a tournament here, we want a tournament back in our hometown. There's plenty of lionfish to go around, so let's do one here. So last year, Ocean Strike Team, which I'm a founder of, stepped up to host a tournament here. We wanted another major lionfish tournament in the Panhandle and kind of bookend. So the start of the dive season, we had the Emerald Coast Open, and now at the end of the dive season, we have the Pensacola Lionfish Shootout. We want to keep pressure on lionfish going into the winter months. One of the things we did see, we'd see that big uptick in numbers of lionfish at the very beginning of the year because nobody really shot towards the winter time. It was hosted by Ocean Strike Team and ran by Gulf Coast Lionfish Tournaments. By the numbers, we removed 2,488 lionfish our first year. We had 27 people participate with 6 teams. Now that's vastly different than what I was used to for Emerald Coast Open. So I keep having to remind myself that this was the first year of the tournament again, and we're not Emerald Coast Open. Maybe one year we'll get there, but it was kind of like starting over for lionfish tournaments. We had one team came from Jacksonville, came over to hunt, and the rest of the teams were local to the Pensacola Destin area. One of the reasons we count for the low participation the first year was we had a really bad weather forecast all the way up from 2 weeks out. Everybody started watching, and it was like 2.5 foot, 4 foot seas down to 1/2 foot sea, then back up to 2.5 and 4 foot seas. And so there was a lot of unknown on what the weather was going to do. And especially with your smaller tournaments that are only that 2-day period, for those of you who are looking at putting together a tournament, weather is a huge factor, and they're going to wait. Divers are going to wait till the last minute to sign up unless you have that pre-tournament period. So they will wait to hold off to see what the weather's going to do. So even up into the day before, we weren't 100% sure what we were going to be able to do. We were limited to 1 day of hunting for the most part. You bailed out, so I don't know what you're talking about. As Eric can attest, the seas were a little rough the first day. And so people went out to the pass and tried and looked out and said, nope, we'll see you at the bar. And so they came back in, but we did get one really good day of hunting. And that was what attributed to our almost 2,500 lionfish with 27 people. So the effort was definitely there. Had we had another day of hunting, we fully expect that it would have numbers been doubled or maybe even tripled. Again, with the weather, the participation was a little bit low, but still first year. I keep on saying that. This is our first year back. So lesson learned in basically starting over again from a place that we were at. After 5 years, this is a brand new event. We did rebrand, we did reorganize. We had a different name, different location. So there is that. But for some reason we thought, oh, we've had this tournament here before. It's going to be easy to get everybody to come back. It's not. I'll tell you that right now. So if it's been a while since you've had a tournament, that long lead time you need for new sponsors and new participants, be sure and watch out for that. We announced a little late because of some of the things that we were trying to get going. And so that hurt us as well. Community support and involvement is huge. The Pensacola Beach Boardwalk Association was such a major help to us, took a lot of load off of what we're doing. Very similar to what we do here in Destin with the Emerald Coast Open, with working with Destin Fort Walton Beach and Destin in the Harbor Walk there, where they handle a lot of logistics on having the event at a certain location. Again, as here in Destin, the Pensacola Beach Boardwalk looked at us more like a partner versus a vendor. And that's a huge distinction. When they're asking you to pay for everything and asking you to do everything and asking you to apply for these permits and do this and do that versus, hey, we got that covered. It's a huge difference on your workload as a tournament organizer and what your tournament participants can be involved in. So it's really cool. Work with your DMOs, your destination marketing organizations, such as Destin, Fort Walton Beach. In Pensacola, we had Visit Pensacola that helped out a lot. Although they weren't direct sponsors of money, they had all the connections. If I needed something, I called them. They were able to open up their partner network to help us out on sponsorships, on things to do, on marketing. Visit Pensacola opened up to one of their partners, Cat Country. So we got quite a bit of free advertising for the tournament just because we engaged Visit Pensacola and got them excited about it. They put marketing on their website about that. They didn't cost us anything, and that was a good help. Lesson learned always, you can't control the weather. Just prepare for it, figure out what you're going to do if you can't have a tournament, and make plans for that ahead of time. Fortunately, we had the festival going on, so the weather on land was great. A little bit windy, but Robert, we sat around and had a lot of great conversations and talks, and talked to the vendors and the participants that didn't shoot and things like that. Just some thoughts I will kind of throw out there and have us think about. Not that it's wrong, but have we created a monster with larger prize pool? I heard a lot in my tournament because we have a little overlap of participation. Several teams didn't compete because we didn't have a large prize pool. Number one, like with every tournament, even Eco, we don't really announce tournament prizes until we've got a good handle on the sponsorship, how much money we have coming in, and how much money we can offer. Same thing with Pensacola Lionfish Shootout. The longer we waited, the less people were like, well, they don't have any money, so I'm not going to participate. In fact, once we did announce it, there's like, that's not worth it. So that was something that we're going to have to fight, and the smaller tournaments are going to have to really look at is how can we incentivize divers or guilt trip them or figure out a way to get them to participate when you don't have a large prize pool. So target maybe more the beginner divers or the beginner tournament people versus the ones that have won the 15,000 or 10,000 or 5,000 prizes in the past. Lean on your experienced hunters to help the first couple of years. We had two teams that had shot many a times, and I kind of, begged and plead, please come to our tournament. Shark Quest is one of them. They put a team in, but despite the fact that it was a low prize pool, one of our other teams that actually ended up winning everything, they're a big participant in Emerald Coast Open, and I just went to them and say, man, help me out. Just throw your weight behind us a little bit. And they talked about it, and they were in that, I don't know if this is worth it, boat fuel and cost and money and time. And one of the team members hit up me afterwards and just basically said, hey, I just put the guilt trip on them and said, if we're ever going to get to a level where we're giving away a lot of prizes, we got to start somewhere. So let's help them out. So if it wasn't for that personal plea of, man, please help me out, it would have been a lot worse, I think. Overall, it was a great year. You take your wins where you can get them. We removed 2,500 lionfish in 2 days from the Pensacola area, and it really set the stage. The Boardwalk Association was very happy with the amount of traffic that came through, so they're extremely excited. They actually reached out to me before I could get back to them to say, hey, let's put the next tournament on the date for next year and on the books. So they're super excited. They're going to throw money behind some more sponsorships and some more advertising. They're getting their people more involved. They're going to push hard on the association side. Visit Pensacola is stepping up again and going to be doing a lot more. So we were successful. Despite the number of participants, the number of fish came in, the event went well. As far as the public concerned, FWC did an amazing job with all of their people that came out to support us. We got the $2,500 for the program that they run, but they also came out and supported us. They had several booths there from FWC. It made our festival bigger. It gave us more things for the public to look at, and overall, it was just a great experience. Save the date, September 20th through the 22nd. Of next year. We'll be back at Pensacola Beach Boardwalk, centered around Bamboo Willie's tournament and festival. It's going to be a great time. This is my personal plea now. please come to my tournament and shoot. We're going to have more prizes, more fun, more excitement, more things to do. I believe that is it for that. Couple of sponsors I want to mention that are here. Dive Pros Dive Shop. They're always a great sponsor. Shark Quest again. FWC. I want to really thank our marine resource team over here for being the account masters and helping out. And I think you've heard an underarching theme throughout a lot of these things, and that's Zookeeper. Zookeeper does an amazing job with supporting our community, and I can't thank them enough. So great sponsors, great friends, decent lionfish hunt. No, I'm just kidding. Great lionfish hunters, and definitely go on a trip with them. It'd be fun to go hang out with them in Curacao and shoot some lionfish.
Try not to hurt everybody's ears here. What's that? Yeah, I'll get it. There's a knob right up here in front. So if the music overpowers you like it did David yesterday, you can turn it up or turn it down.
Gotcha.
And I will hide this.
Can you answer a question? I haven't heard anything about giving back to the community. A lot of your big tournaments, like, Destin Fishing Rodeos, Pterois, they have like community organizations that they give back to. So is that just not something that that is not in the plan? Am I wrong on that? I just noticed all this coming in and all these prizes. I'd like to see like what impact in the community organizations.
So we don't necessarily give back to community organizations. We give back to the divers who are removing the invasive species. The whole premise of the tournament is to remove lionfish and to incentivize divers to do their own conservation efforts by removing the lionfish. Now, what we would love to see, and I think you've participated in the past, is have you involved in our tournament, our festival, so that way you can, for lack of better words, and don't take this the wrong way, ride our coattails into a large festival where you can push your initiative there and that way and push getting your volunteers and getting your message out. That makes sense.
For the Emerald Coast Open though, Harbor Walk Village is where we host the tournament and the festival. They do sell merchandise and everything that they make off of it goes back to local nonprofits. So I, don't know how they go about picking those by any means, but it does go back to local organizations. So.
Education and outreach.
All right, without further ado, I'll introduce Brady Hale. He's the founder of, my name is Brady Hale. On this side, I'm the founder of Ocean Strike Team. We're a nonprofit that does education and outreach as well as research and action. Since you've already heard me talk a lot, there we go. All right. Since you've already heard me talk for about 15 minutes, I'm just going to play a little video. Is there a mouse?
Ocean Strike Team is a nonprofit that helps ocean lovers get involved in marine conservation efforts. Our name actually comes from our motto. Wherever the ocean needs us, we send in a strike team of volunteers and citizen scientists to help her. We follow 3 tenets to guide us: action, education, and research. Let me show you where your money goes when you donate to Ocean Strike Team. For almost 2 years, we've been involved in removing invasive lionfish from our oceans. We teach divers the proper and safe way to hunt lionfish through hands-on, actually in the water training programs. We organize hunts and we take divers out to lionfish hotspots and remove them. And that gives our native fish like grouper, snapper, and triggerfish a chance to grow up and get big enough to not be preyed on by lionfish. We also conduct non-invasive shark surveys. So we dive for sharks, and as they swim by, we have a setup with 2 lasers and a GoPro camera. We video the laser dots on the shark. And because we know the exact distance of those 2 dots, we can use that video to measure them
It determined things like species, sex, size, and then also document a whole host of scientific data. This research is used to give scientists, conservationists, and other policymakers hard data to make informed decisions about the issues related to the sharks and then quantify their important impact on our oceans. We work with other state and national agencies to assist them with projects. Under research permit with the National Park Service, we monitor and remove lionfish in Fort Pickens National Seashore. And we work with Florida Sea Grant with monitoring and restoring seagrass in our local Pensacola and Perdido Key bays. We actually just started another program with NOAA to conduct water quality monitoring, specifically around our local oyster beds, looking for the presence of harmful algal blooms, such as red dye and toxic algae. We lead and participate in beach cleanups to stem the flow of trash and plastics before reaching our oceans. Plastic pollution is a serious problem. It takes everyone doing their part to clean it up. Unfortunately, lots of trash does end up in our oceans, so we also conduct underwater marine debris cleanups with divers on our local wrecks, artificial reefs, and natural bottom areas to remove it before our sea life dives and try to consume it. Education is key to our conservation mission. You have to get the public engaged with the issues. You have to help them understand why we do what we do and how it affects them. We set up booths at major events and talk about plastics in our water, the problem with invasive species, and other issues that we tackle. You have to help them fall in love with the greatest resource in the world. And when that happens, it's much easier for them to want to care and do something about it. This year has been a momentous year for Ocean Strike Team. We have grown our volunteers and membership to new levels. We purchased a boat that will give us more opportunity to conduct more campaigns and more trips. We've added amazing leadership that will guide us into the future. We are absolutely a team, and it takes all of us. And that includes you. We are a donation-funded organization, and ocean conservation is not cheap. Boats are expensive, diving is expensive, fuel is expensive, research is expensive. We just can't do it without your help. But we have big goals for this next year. We're working on getting our boat up and running. We're so close, but she still needs some work. Once we're able to run our own trips and campaigns, the sky is the limit on what we can accomplish. Well, that or bank account. This will give us the ability to run more lionfish trips, which means more lionfish removed, more shark surveys, better water quality monitoring, and so much more. It's a game changer for our organization. We're developing an internship program and citizen scientist certification. This will provide online, classroom, and in-the-field experience for college students and interested citizens alike to learn scientific skills that they can use here and take all over the world and leave them. With your donations and support, we can continue to make Ocean Strike Team a true force in the marine conservation world. We can give our oceans and our community the help it needs to be clean, be repaired, and be healthy. The ocean is calling for help. Will you answer? I'll be over here taking checks and credit cards here in a few minutes. Yeah. I wanted to just kind of show that video, kind of give you an overview of what we do. We cut our teeth in the lionfish world. That was the first thing that I got involved in and started organizing things around was lionfish hunting. All right, so when it comes to lionfish hunting, we kind of have this philosophy. Give a man a fish and you feed him for a day. Don't teach a man to fish and feed yourself. If he's a grown man, fishing is not that hard. Lionfish hunting is not that hard, but sometimes it does take somebody to just kind of show you how to do it. When I first started in this way back in 2013, I came down to the first Lionfish Removal and Awareness Day just to see what was going on, learn about lionfish, see what was happening. I was working a dive shop in Dallas and we had lots of questions. So I said, well, might as well come down there and find out what's up. I grabbed my dive buddy who's pretty adventurous and said, let's road trip it. That year, the tournament got blown out. And so I got to meet Alex and Andy and Ali and Keith and all the Dan Eleanor and all the bigwigs of the lionfish world at that time to really find out what was going on. And they kind of issued a challenge to me coming from Texas saying, what are you guys doing about it over there? And the answer is we don't do anything. Absolutely nothing. The dive community didn't know about it, didn't know about the problem. So they said, well, somebody's got to do it. And so the 9-hour trip home, my dive buddy and I kind of kicked that back around and said, well, you know, we already have a stage with the dive club and we're already doing talks about this stuff. Let's figure this thing out. And maybe we can give a talk over there to our dive club. Well, I ran in, you know, I took a trip with Captain Andy and I was kind of hooked. He really showed us the ins and outs of lionfish hunting and how to do it. Went on his boat, shot 90 lionfish the first dive. And man, I said, I'm in. This is awesome. So then we started to come back every month. I'd bring another person, and next thing I know, I'm calling Andy saying, hey, I need the whole boat for all weekend long. And that grew into this organization that we have now. 5 years ago, we started getting into shark research and working with the no fin Florida bill. That's where we kind of diversified out. But when it comes to lionfish hunting, we run ecotourism trips. So we have a 3-day program that we run with a little bit of classroom and a whole lot of lionfish hunting. It's agency independent. So whether you're PADI now, TDI, SSI, you can all come. We don't give certifications. We just run a good quality. We're going to teach you how to hunt lionfish. You don't have to have any equipment. We provide the pole spears. We provide the zookeepers for you to borrow. And what that does is it gives them the opportunity to come without having to get anything else besides just their dive gear. We're going to supply everything else, and we're going to show you how to do it. And we're going to send you away with the hunger and the skills and the knowledge on what you need to do on your own. One of the biggest measures of my success of this program was about 3 years in, when I had a whole group of people from Tennessee, and I didn't know a single person on that trip. It was all just done through marketing efforts. They found us. So they came down, and about 3 months later, I got an email from a couple that was on the trip, and he was the lionfish hunter. She wanted nothing to do with it. Then she started pointing them out for him, and then, well, let me just try one. And the next thing it was like, get out of my way. This is my fish. They were in Grand Cayman, and they sent me a picture of like 15 lionfish on a deck. And they're like, we did it. We got it. So they went from not knowing anything about lionfish hunting to being able to go to a foreign country, know what equipment to use, know what gear to use, what skills to use to actually remove lionfish. So that kind of shows that ripple effect of when you teach one person through education and outreach, they're going to have effect worldwide, basically, when it comes to this. So this is kind of just some images of all the different groups. We have all ages. The kid up in the far right-hand side, he's from Colorado. He was 16 years old. He was doing a piece on, I think it's a science project on lionfish in Colorado. Never hunted lionfish before. His mom called and said, hey, is there any way we can get him in the water? He was 15 because there was some restrictions on depth. So I laid out for him, well, you need to get advanced. You need to get deep diver for juveniles, whatever it's called. Then we need you to do so many dives before we can take you out into 80 to 100 foot water and shoot lionfish. Well, they called me back and said, all right, we're there. We're ready to sign up. So they flew down from Colorado. 4 of them flew down from Colorado to learn about lionfish, to then take that knowledge and experience back to Colorado. So pretty interesting stuff. So that's what I want to talk about as far as the lionfish outreach. That's our main part of that, is to teach them how to do it and have that ripple effect of what's going on. Any questions? I got time. Cool. Time for questions? Hearing none. That's my contact information. By all means, feel free to give me a call. I can help you in any way that I can. Like we saw on the slide on the video, we're involved in a lot more than lionfish, and I've already talked to a couple of you about doing some projects together. We do operate as kind of fins in the water for a lot of researchers. If you need lionfish for projects, or you need volunteers to help go do things on your projects, especially if you have funding, then we'd love to help you out and get some divers and volunteers and ocean conservation lovers involved in what you're doing as well. Thank you very much.
All right. Thank you very much, Brady. Was that picture from the other day?
That was.
Yeah. All right, now we have Stacy Danko. She is the best marine science teacher over at Fort Walton Beach High School. So she's going to talk about I don't know, educating our students about lionfish, I guess. Yeah.
All right, hello. First of all, there's no way I would be able to have my program at Fort Walton Beach High School if it wasn't for Alex and Jessica. So thank you.
Microphone, please.
Oh, sorry. I know, I'm short. I know, I need to put down the microphone. Vertically challenging issues. So first of all, everybody at Fort Walton Beach High School calls Alex the lionfish slayer. They don't even call him by Alex anymore. It's like, when's the lionfish slayer come? And then Jessica, they love you. Every single time they come, they're like, when's Jessica come back? When's going to do this? So this is a program that my students look forward to every single year. And so I'm very, very happy to have a lionfish program. So a little bit about Fort Walton Beach High School. We serve a lot of the Emerald Coast, Destin, Fort Walton, Mary Esther, Eglin, and Herbie. We have about 1,800 students here and there. And the course that I teach for marine science is junior and senior. And I actually now teach AP Environmental, which I'm way excited about. And that is 9 through 12. So yay, I got the two best classes to teach ever. And so how do I integrate lionfish education? It addresses so many Florida Sunshine State standards for AP and for marine science. It's most definitely STEM. And the best part, I'm not going to get into too much education and pedagogy, but definitely 5E. It's engagement. It's awesome. Kids love it. And you can see here, we have Chef Jim from Dewey Destin Seafood come cook lionfish every year. And like students like literally fight each other to come to the desks first to grab fish. And they absolutely love it. So we call the day Lionfish Day. You can see we have a host of students up there. We had a live lionfish here and there, and it is, we have to tell them, you may not put your hands in the tank. If they do, they're going up to the office immediately. But this really does alleviate a lot of misconceptions that students have. The first thing they think, oh my God, am I going to die by eating a lionfish? And the answer is no. We explain how, one, to fillet them, how no, their meat is not poisonous, and they use the word poisonous all the time. So we have to differentiate between poisonous and venomous, and then they kind of get an idea of that. And then we also talk about the invasiveness of the species. We have them do a whole project and make them make wanted posters. We do little wheel names. They can choose what they want to do. And the first one that every student chooses, lionfish. So students already know what the lionfish here. It's just a matter of, okay, how do we get these misconceptions out there, correct them on them, and get them very excited about lionfish? So one, they're not scared to touch them anymore. After we explain to them, you know, about their spines and how the heat denatures the venom, they are more than eager to come up. All right, let's touch them, let's play with them, and obviously eat them. Chef Jim does a beautiful job of showing students how to fillet them, and it just makes the students feel so much more at ease. And this picture here, I love just because you can see everybody just surrounding the desk. All of the students are getting along. They're not scared to come up and approach and learn so much information. I love it. It's just, it makes me happy seeing all my kids all the time. And yes, I call them my kids, even though they're students. And so this is how Chef Jim comes up here and he makes these little fillets. And I really wish my students understood, and I hopefully they do, on how unique this program is. You don't get people coming in here and cooking lionfish for you, and the students literally, they devour it. And then I have teachers coming in now. Oh, it's Lionfish Day today. And they find my classroom's way in the back, which I like. The teachers find their way back to my room. It's like, I haven't seen you in 5 years. Now you're in my classroom wanting lionfish. I see how it is. So hopefully you guys kind of understand how this really does engage the students. They ask so many questions. After Jessica and Alex leave, it's like, I think they're scared to talk in front of them sometimes. And then the next day, ding, 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 question, question, question. And they want to always learn more. And so I have two of my students who couldn't be here who wanted to speak about it. First of all, Joanna, she's so tiny and she's so quiet. And she's like, I want to help you do your presentation. So she's very nervous in her video. So kind of be a little grace on her video, but she did it. and I love her too. She's so sweet. Where's the mouse? Space bar. Thank you. Oh, wait, it went to the next. Ah, there it is.
Hi, my name is Joanna Lee, I'm a senior at Walton Beach High School.
I'm gonna let you play with it.
Very excited class where we got to eat lionfish. I'm usually really picky about seafood. I tend to find the texture like weird and like off-putting. But then the day came where we actually got to eat lionfish, and it was genuinely the best thing I've ever eaten in my life. And it was very light and flavorful. And I'm not even exaggerating when I say I told my mom all about it, and I told her how she had to try it. And with how invasive they are here, it would be a great way to control the lionfish population. And afterwards, we could follow the footsteps of other places that have taken the spines of lionfish, removed the venomous barbs, and turn them into jewelry like earrings and necklaces. And it's also possible to take the skin of the lionfish and turn it into leather, but it is more expensive. But with the jewelry, it could create new economic opportunities.
So this is a perfect example because they're already figuring out how do we make this more marketable? What are other things that we can do to allure people? Okay, we're going to eat the lionfish, but we can use the other parts of the lionfish too. And it'll also help raises awareness. I'm so proud of her. And then the next student, we just call him Bailey. I love him to death. He wants to be a lawyer. And when I said I needed help doing this, Bailey was the first one to jump up. I got you, Miss Danko. He's very, he's got a personality on him, and you'll see in a second his personality, and I love him.
American marine biologist and oceanographer Sylvia Earle once said, "The more we learn about the ocean, the more we know about ourselves." And when I began learning about lionfish in my marine science class, not only did I begin to understand these creatures, but also about how human influence is the key to solving the problems caused by them, as well as how human actions have played a significant role in their detriment. The integration of lionfish and marine science in my school has helped me to learn more about these majestic and tasty creatures than a textbook ever could. When we watch informational videos rather than skimming lines of a textbook, when we get information from people on the scene dealing with this issue like Miss Jessica instead of staring aimlessly at models of these fish, when Miss Dingo invites Chef Jim to not only display the anatomy of these animals, but to also show us one of the best ways to delay their invasion, it develops a form of learning that does not remain in the classroom. At one of my debate competitions last year, I was given a topic, and I had to give a speech on something completely random. But because of how in-depth and innovative lionfish was integrated into my marine science class, I was able to give my entire speech focusing on the invasiveness of these beautiful creatures and how we can stop their infiltration. The goal of education is not to prescribe memorization, but rather to instill a yearning for knowledge. And I am proud to say that because of all the unique ways lionfish has been embedded into the curriculum, my strive for knowledge on these creatures has not ceased, but rather has led me to do a deeper dive. And while I may not be the Sylvia Earle of lionfish, the importance of educating marine science students on lionfish is one we should all know more about. Because the ocean is many things. And it's because of Miss Dingo, I know what part I can play in helping it thrive. Thank you.
And I really can't top, I can't top him. I can't. And he references Sylvia Earle a lot because I want to be her because she's amazing. And almost all the time I'm saying something about Sylvia Earle and he's like, oh, I got you on her. So I really am going to wrap it up with Christian because he did such a fabulous job. I invite anybody, eat the lionfish. You guys know about it. You've probably eaten it. It's wonderful. And once again, Alex and Jessica, thank you so much for coming into my classroom and helping me make Fort Walton Beach High School the best marine science program in Okaloosa County. Yeah, does anybody have any questions? microphone. Does anybody have any questions right quick? Okay, we're good. Merci.
Thank you.
All right, now to wrap things up, we are going to have Charlene Morrow up here. She is going to talk, well, she is from the Navarre Beach Marine Science Center. And she is going to be talking about lionfish education as well. So have it, Charlene.
Awesome.
Thank you. Well, first of all, I really appreciate you guys including educators in this workshop. It means a lot to be here. And thank you to so many of you guys in this room of giving me the opportunity that I have to, introduce my students to pretty amazing stuff. So I want to tell you, first of all, you might ask, I didn't even know there was a station on Navarre Beach, but just a little background information. I teach dual enrollment, marine biology and oceanography to students from all around Santa Rosa County. And they come to me 5 days a week. And 3 days a week, I teach them marine biology and in the spring oceanography. And then 2 days a week, they teach area schools children that come out. And that's how we generate funds. A lot of misconception is that, oh, you must get all school district fund you. No, actually, it was totally up to me to convert this station and, with lots of help from the community, raise the money and write some grants and make it happen and show it's sustainable. And that was going on 15 years. So even before the station opened, I started the marine science program at Navarre High School. And to show you how much I like lionfish, you see this picture right here. This was about 20 years ago. I had lionfish in my classroom, and I guess there was a disgruntled parent that saw this article in the newspaper, and I got in trouble. So I wasn't allowed to have lionfish, but to be honest, I have one again. So anyway, I know I'm such a rule breaker, but gives you an idea about what our students do. But what I want to kind of talk about a little bit today is how we're a combination of formal and informal education, and how I take what I teach my students, how they use that to give back to the community and do these festivals and ideas. And it's pretty amazing. So this is kind of what I'm going to go over. I'm going to talk a little bit about what I do with my students and then how they work with younger kids, what type of programs we have that relate to lionfish education, and then how we've been involved with the LRAD, the festival, for 10 years now. So in my class, as far as lionfish, yeah, you guys come out, Alex, guest speaker, Robert, guys, Rick, all you guys come, and I appreciate all your help. And I hope the students appreciate it too, how much you guys take your time and come in and see these. But I think it's paying off too, all this extra stuff. So this was one of the projects we were involved in like 7 years ago, where our students, it was a Will Do grant, Gulf Breeze Will Do. And we worked with UWF, and the kids actually did their own DNA barcoding. They did the whole process, gel electrophoresis. And it was a learning experience for everyone, but it got a lot of kids into it. And just to give you an idea, well, one of my students, I have 2 students here, and I didn't make them come here. It's just that Katie is in the back, and she was part of this project, and now she works with you guys. So that's awesome. And JT is one of my current students who's really into drones and ROVs, but I will not make you talk in front of everyone, JT. So that's our unit. We, teach, of course, a fishes unit and so on, and then we do the lionfish. But what's cool is that our stations that we teach to visiting school groups, my students have a lot to do with creating them. And so I want to show you a couple of the things that we do. Our kids, and I know this, they're gonna be like, why does she have this? One of the things that we participate in is the Sea Perch program. I've got props, right? And the kids get a kit that they create their own ROVs, they do their own soldering, and they look for a real world application. So they're in teams of 2 or 3, and they have to do a presentation about their topic that they want to use their little kit to create a working ROV. They do it all. I kind of just give them the kit and be like, have fun. And they already know how to do a lot of this because they did robotics in like middle school. But yeah, this one has a name, it's Rug, and it's designed to catch lionfish, which I thought was kind of creative. And I had mentioned to the kid, no, I know it's not something that you're going to go out and actually use, but it's conceptual. And it got the kids thinking, I mean, look at the design on this ROV, it's flat, huh? So they actually try them out. They show the class when they talk about why they came up with that design. And instead of spearing a lionfish in our little pool that we practice in, they did a balloon. So that was fun. And it was amazing that these kids come up with these ideas. Like we had one group that did an ROV with like for a midwater trawl. And then they take all that, they win regionals, hopefully. And then last year we had a team that went all the way to international ROV competition and won. That was 70 teams from around the world. And we had an incredible engineering group minded, and hopefully we'll do it again. And by the way, my number one volunteer is here, and that's Carl. Carl is not only our dive instructor, and he helps with anything, I mean, everything at the station. He also is a mentor to those teams that compete at nationals. But another thing you can see here, you probably recognize that little lionfish toss, you know, these guys. By the way, we used to have these at the festival in Pensacola, and then turns out they're collectible, and now they're worth $50 each, which is crazy. But we've been throwing them all these years to knock out critters, and but I had some other ideas for these too. But this kind of gives you an idea about our school groups. I am just a facilitator. I teach these kids, they teach younger students. And like this activity right here is about investigating sand. It's called Sand Sleuths. So there's mascots that, you know, there's a mystery on where the sand came from. And man, I can't tell you how rewarding it is to see these kids do the teaching. But up here in this picture, right up here, I don't know if you can really see it, but speaking of lionfish, there is a paper activity that I think it was Dolphin on Sea Lab that came up with it, that you stuff a paper lionfish and you have a stomach with like 17 different types of fish inside of it. And I was like, oh, you know, that's kind of cool, because they'll take them out, they'll identify the little fish, and then they will, you know, do percentages. So it's neat activity. And my mind's like, oh, okay, what can I do to make this even better? So and we're going to do this with a school group probably, but I tried it out with my students. So I took these old Webkins, and then instead of like paper going inside, I created these little stomachs. And you guys could do this. I know some of you need like a broader impact when it comes to education, maybe in a grant. You might want to. And so I have all these little fish. Oh, there's a seahorse. Oh, there's a blue tang. And so the kids would take these little parts. It might be a shrimp part, and they would use a data sheet and then figure out what's in the stomach. And I'll even shove a little lionfish toy in there, maybe some nurdles. I don't know. So I think it'll be fun. I'm just trying to make it more and my students help create this stuff, which is amazing. So as far as community involvement, this is a pretty popular program that I think that other, tourism groups might want to look into it. But we do these snowbird sessions by the sea. We've been doing them for a while. And once a week, we would have a different topic. So as you can see on the schedule, one of the weeks is lionfish. So sometimes we have a guest speaker. Sometimes it's just my students talking about it. But at the end of this 1 hour program, we fry up some lionfish fillet. Thank you, Carl, for getting those lionfish. And so these, look how they love it. These adults, at first I was like, oh gosh, I don't know about teaching all these adults. And they are just like my students. They love to learn. And so there's one, look at coastal ecology. I think, Rick, you came for that. Beachcombing's a fun. These are all programs that, appeal to everyone, not just, they want to learn too. And so it's been great. 10 years. First Lionfish Festival we did, I think, in 2015. Yes, that VW Best, I had great plans for that, but it was really a rust bucket with a really cool wrap, and no longer have that. But yeah, I really liked, and thinking about this festival, I really liked how it was, people that came to it they're there for the lionfish. I liked how the chefs were involved. I know that that's not, it's pretty packed here in Destin, but lots of great memories from this. This is where we first met Guy Harvey, and it led to the group sponsoring our fishing camps that we have or did have. And here's our setup over the years. And that is me at the Lionfish Festival a couple years ago. I had a custom lionfish mascot made. So yeah, thousands of dollars in the US. You can find someone in China to make one for like $500, and it's a win-win for everyone. So I should have brought that today. And just where's it going from here? We're going from touch tanks, and what we're seeing, what we're doing in our booths more often in the past couple of years is the dissection. So we get the fish that after they are measured and so on, when they come in from the tournament, my students will go over and get them, cut off the spines, and then dissect them for people that want to learn a little bit more about the lionfish. And some of them are really grossed out, the people that come up to them, but that's why we have we can do a simulated dissection for those. But yeah, I look forward to working with the group for many more years. And just the little idea for those of you guys that are into sponsoring, we do a Autism Odyssey event, and it's for almost 200 families with kids that have special needs. And then this year we are expanding to, we're going to have more adults at the event, and it's all organizations providing sensory activities. We take the kids paddle boarding and, fishing. I think you guys have had a dive booth there before. So it's a special day. Maybe you just want to volunteer. Just let me know. And yeah. So what is next? Keep doing what I'm doing in the classroom, and the time that you're taking to come into my classroom and involve these young kids at these events pays off. And what's really cool is Kristen, the one that worked under Will Patterson, that did the lionfish. She was my student 20 years ago. And so yeah, we're investing in these guys, and they're going to be where we're at right now. And yeah, thank you. That's it. Oh, are there any questions? Oh, come on. Yes.
For your students, are they coming after school, like as a extracurricular?
Yep.
Or it's like summer camp based?
As part of my curriculum, they have a requirement of 15 service learning hours outside of class. But on average, they're doing about 30 hours a semester outside of class. So it's a commitment, but at the same time, I provide a lot of opportunities for them as well. Any other? Thank you.
Thanks a lot, Shirley. All right, guys, it's time to wrap it up. First, I would like to thank everybody for attending. I really appreciate all the presenters both days for presenting all these research projects and other education outreach projects. I would also like to thank Alex and his team for putting on this thing, Aramark for doing this. I think the event and the venue is awesome. I thought the food was great. I really appreciate you guys helping us out with that. I also like to thank again our steering team for helping us put together the whole program. And again, as I said, I would like to thank you. Hopefully we got a lot of education out of this. When we put this idea of bringing this thing back after a 5-year period, one of the things we wanted to learn about was the research that had been going on in lionfish, what has happened over the last 5 years. Because as you just heard, a lot of us are involved in education outreach. Are we given the best, most updated information? And some lessons learned real quick with Will's talk on the population and how things are going. We know in 2013, we had some of the highest densities in the South Atlantic region. In 2018, that had dropped. And what we learned today was that basically it's still down. There's some anecdotal reports that it may be climbing back up, but right now, and it could be linked to the disease, the ulcers. That was another thing I was really interested in hearing today, was any updates on the cause of the ulcers. And as we heard, it's still an unknown. but the fish are out there. So it'll be interesting. Again, we'll do this again in 5 years and see if there's any updates on any of that research. The eDNA, the Dr. Linsky, that was awesome. That was totally awesome. You know, to hear about we'd heard about this in the upper estuaries, but the whole thing on the shorebirds was something that was very cool. So again, we'll find out in 5 years what else and what other targets might be with that eDNA work. That was awesome. And then of course the PCB stuff. I mean, you know, again, we got focusing on the lionfish. The PCB numbers are down. We'll see how long you still have to run it, but again, it's a target effort now on lionfish. And again, we'll get an update on that, whether you're still doing it or not. I know you guys will be giving an update on that. The harvesting, you heard what we heard from the seafood markets over there, what Thomas and I brought up. And again, any feedback from you guys that we can take to them to see if we can get across those barriers. But again, yield and number, they've got to clean in a period of time. And then they're still a little concerned about the venom in the spines. So we'll have to do some education. We will be meeting and talking and having a program with them this year. So we will definitely follow up on that. And with the tournaments, it sounds like everything's going really well with the tournaments. They're getting a lot of turnout. The numbers are good. We did see a little bit of kinks with the commercial side, which was interesting that went along with what we've seen on the commercial side for the harvest part. And then the large prize part of it, you know, how much of that has got to be part of a tournament to make it successful. But it sounds like the tournaments are going pretty well overall. And then the education, I mean, it's just crazy awesome what you guys are doing with these kids. I mean, the student presentations are awesome. The fact that you guys really have lionfish specific stuff going on in class, I think it's great. And then of course the citizen science that Ocean Hour is doing and a lot more. And one of the things she found was eDNA evidence of the fish in the bay. Now we want to put some eyes on it so we can get ocean strike in the water and maybe some ROVs along the bridges because you can't dive there anymore. And find out if the fish are hanging around the bridges there in the bay. That would be kind of interesting. But again, I would really like to thank everybody for participating. Do not forget, please, please, please do not forget to complete an evaluation and put that in the tan tray on your way out. And thank you. Thank you for coming.